the Father. As usual, remember to be positive, optimistic, keep positive people in your circle, as those three things are key to ensuring positivity is invited into your future. It is February 16th of 2022. And it is 3 o'clock p.m. Eastern time. I'm located in Baltimore, Maryland, for those who don't know. And I'm going to go ahead and start what we have called the DNA lecture, right? For anybody who didn't get a chance to watch this or come into the live, I would recommend that you watch it back if you get a chance. This is for the purpose of us all having at least a rudimentary understanding of what DNA is. You know, if the term deoxyribonucleic acid bothers you or makes you like, what is that or whatever, you definitely want to watch this video, at least so you can familiarize yourself with a lot of the different terms and what they are, especially if you're somebody who worried about the jab. You know what I'm saying? Because this is things that you should know about. After we finish this lecture, you're not going to have questions about whether or not cloning is real. You know, after we finish this lecture, it's not going to be weird to you to think that they could modify, you know, certain things about living species. Peace to you, Reflection. Shout out to everybody in the let me wave at everybody. I want y'all all know I love y'all and I appreciate y'all for coming into the live. You know, so I'm gonna jump into it, but like I said, before I jump into it, I just wanted to say to everybody again, I appreciate you if you watching this. If you get a chance to watch this back, I send you nothing but positive vibes, because it takes a lot to take time or energy out of your day to interest yourself. Peace, Reflection, Pharaoh. It takes a lot to put energy into something that's never really been of interest. You know what I'm saying? Like, I never had an interest in chemistry or DNA, but this is something that we got to do, especially if you're a parent. You have to know these things. Like, you can't just be ignorant. I mean, you can, but I wouldn't recommend it. Okay. All right. So first off, DNA, right? DNA. And the whole purpose of this, right? The whole purpose of this is so that these terms, let me make sure this can, hold on, y'all. Hold on. Give me 2 seconds. Get this camera clear. To make sure that this structure right here is not foreign to you. So you don't look at this and go, what the fuck is that? Right? Because normally when you look at DNA, you like, what is that? All right. So let's just break something down before we even go into it. Deoxyribonucleic acid, right? Deoxy refers to oxidation. It refers to the removal of oxygen atoms, right? So anytime you see deoxy in front of something, just keep in mind that they're referencing the removal of oxygen atoms during a chemical process, which is normally something like hydraulics or hydrolyzation. Fuck their words. You know what I mean? It's a term that they got for where they remove oxygen atoms. All right. Shout out to Indigo Star, one love to you, Reflection, everybody who coming in. All right, so that's deoxy. Ribo refers to ribose, right, which is a sugar. We're gonna go into all of this, but I'm just getting the tone set, right? Ribo refers to a sugar, which is known as ribose. It's a chemical base for DNA, right? Nucleic refers to the nucleus inside of the cells, right? So they're saying basically at a quantum level, they're basically saying oxygen removed from sugar in a nucleus, you know, in the nucleus. That's what this is saying. Oxygen removed from sugar in the nucleus, and then acid. For those who don't know, acid is basically a substance that can donate protons, right? And that's why it eats through shit. So like normally it's basically capable of donating protons to anything or removing electrons to anything and giving it a charge. It depends on how strong the acid is. There's different types of acid bases, and we'll go into those if I come across them. Anything for people to know, just so that y'all know, before I jump back onto that. As we go through these, I'm going to go through the links as I see fit, and I'll go through it as much as possible. But, you know, it would take us all day if we went through all of these, because just understand that every single link you see here, every one of these is a whole lecture in itself. You know what I'm saying? Like, to be honest with you. But let's just go into it. All right. So DNA stands for deoxyribonucleic acid. For those who don't know, RNA just stands for ribonucleic acid, right? That's before it's deoxidized, right? So let's see. Deoxyribonucleic acid is a molecule. Now I'm going to make sure I go into these so y'all can all know. Yeah, I'll save the live reflection. A molecule is just a group of atoms. Let's see what they say though, because I want to make sure that you understand what these things are. So a molecule is an electrically neutral group of two or more atoms, right? So just keep in mind that, and you're going to see a lot of these words used interchangeably, but just know that a molecule is a group of atoms that have no charge. So they're neutral, just molecule, right? And it's a group of atoms. All right. So DNA is a molecule composed of polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses, right? So let's break this down a little bit. Polynucleotides. Now, a lot of things y'all want to keep in mind is that in chemistry and in science in general, you're going to have a lot of the building that has the door in it, that has the door to the apartment, that has the room, that has the closet. You know what I'm saying? You're going to find a lot of these. Like how you have an atom, and then the atom has protons and neutrons, and then protons and neutrons have hadrons. Like, it's just like a breakdown, right? So when they say polynucleotide, keep in mind, poly refers to many, right? So it's normally contrasted with mono, right? So keep in mind, they're just saying many nucleotides, right? And nucleotides are what the backbone of DNA is made from, right? We can click on it so we can see. We're going to go into these in more detail as we go along because it's a whole lot to cover. But I just want to show you. Polynucleotide molecules is a biopolymer. There go that word again. Now, bio means life. It means living organism. So anytime you see bio, just think of living. Polymer is basically a group of mers, right? And I know that sounds weird, but mer just refers to a link in a chain. So when they're saying biopolymer, they're saying a group of living organisms that can be chained together. That's what biopolymer means, essentially, right? So a polynucleotide molecule is a biopolymer composed of 13 or more nucleotide monomers. Remember what I just told you, see? Monomer. Monomer means one of these. A polymer is a group of them bonded together. A monomer is one out of that chain. So let's say you had a bunch of like hydrogen atoms chained together. One would be a monomer, right? So basically they're just telling you that the nucleotide, let's go into it real quick. It's just telling you that it's a bunch of these chained together. We don't have to go into it like that because it's going to make more sense. Let's not go into it. So just know that this polynucleotide chains, they're basically talking about these. These right here are called polynucleotide chains, right? And we're going to get into what polynucleotides are because there's 4 bases that make them up. Let's just move forward. Another thing I wanted to show y'all, this is a very important thing, double helix, right? This is where sacred geometry and biochemistry come together. You see, because when you go into double helix, let's see how much they'll show here. I don't know. I know we're going to encounter it later, right? So, and I'm going to go into this too, but I just want to read this here. In molecular biology, the term double helix refers to the structure formed by double-stranded molecules of nucleic acids such as DNA. The double helical structure of a nucleic acid complex arises as a consequence of its secondary structure, right? Now, they're basically just saying this line, this is a double helix because this line is adjacent to this line, which makes it double helix. That's all I was saying in complex words. But let's go into something because I want to show y'all something. There's multiple types of DNA for those who didn't know, right? We could read all of this, but forget all of this. It's just telling you, you know, the structure, why it's shaped the way that it is and everything. And there's a reason for that, which we're going to go into in 1 second too. But peep this. The DNA double helix biopolymer, right? Which is a group of living organisms, a group of living, molecules, right, of nucleic acid is held together by nucleotides, right? Nucleotides are biopolymers, basically, which base pair together, right? Now, bDNA, this is what everybody's familiar with, bDNA, right? But there's multiple types of DNA, which I wanted to show y'all real quick. So check this out. You got aDNA, you got bDNA, which is what everybody know. This is right-handed people. This is the norm, right? And then you got zDNA, which is left-handed, right? Now, when they say base pair, right? So right-handed, the only time they'll tell you, and we can read up on it, but they'll tell you, just trust me when I tell you, you can look it up yourself to confirm, but aDNA, this is something that happens anytime there's some sort of a outside influence on your DNA. So say like you suffering from hypothermia or something, you burning, something is crazy that's causing adrenochrome or adrenaline to be running through you. Then you have this, right? This is not that common. But just keep in mind, these, when it says 1BP, this is talking about base pairs, right? And base pairs, which I'll read up a little bit on so y'all can see. I want you to know the importance of that, because look at what left-handed folks have. They have double base pairs, right? Now, keep in mind, all of this is his story, so they could be lying to us about all of this, but I'm just letting you see what they tell you, right? And by the way, for all of the Wikipedia gurus out there who like, no, Wikipedia is evil and blah, 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 here's the references for this. So you can highlight this and it'll show you whatever white smart guy you want to tell you so that you can feel better about it. Rich Norham Wang, chemistry. He's the chemistry and biology. He wrote a book in 1984 called The Chemistry and Biology of Left-Handed zDNA. If you can't learn from Wikipedia because of whatever phobia you have about how it's fake, go read this book and then you'll feel better about it, okay? Same thing for this one. Go read this, all right? For all of those Wikipedia haters out there, go read these books so you'll feel better about what you're learning. All right, so back on what we're talking about. So two base pairs, and base pairs just represents, this is what they look like. Look at what they look like. So this is an A. See, this is the one we used to, right? It just looked like normal double helix structure. But this is the A. See how it look all afflicted and messed up, right? But then look at this one, the Z. So the things you want to keep in mind, and I don't want to make this too long, y'all, my bad. I just wanted to show y'all these differences. The base pairs refers to these meetups that you have here. We can see it better on the other screen, but it refers to where they have the hydrogen bonds. When they bond and connect to each other, they have two. So left-handed people have two for every repeating unit. So instead of it just being one base pair connecting them, they have two of them. That's exquisite. Like, that's something that you got to really pay attention to. And one love to everybody who's in the live, sending positive vibes to y'all for actually watching and getting information rather than watching twerking and hip hop and loving hip hop and shit. But look, this, like I said, this is important. Another thing, the rotation of the BP, right? You know how y'all see it on the diagram screen where they be spinning and it just spins like this in a rotation. That's representing the circular motion that our DNA travels in. This tells you how much of a rotation in degrees it gets. So one rotation is 34.3 degrees for normal DNA, but for left-handed it's 60 per 2. So 60 for every 2. You know what I'm saying? So basically, yeah, they saying for both, for the base pairs, yeah, because it's 2 of them. I was like, why are they saying divided by 2? But yeah. So then keep that in mind, right? So that means it's spinning faster, so it's able to send information faster. And then the last thing you want to focus on is the diameter. And the diameter, this is like a fancy word. You know, they make up these big words because they want it to be intimidating to you. What's up, Diamond? I appreciate you for coming into the live. We're talking about DNA and biochemistry. If you want to know about your anatomy and what you are, check it out. You know what I'm saying? I'm putting it in the regular terms. The diameter just means how far apart it is. So like if you make a circle, the diameter is how far it is from here to here. That's all diameter is. This is nanometers, right? So nanometers is a molecular, it's a molecular measurement of space, right? So you see the right hand is 2.3 nanometers. Now, every nanometer matters because that's how quick information can get through. And as we know, like with computers and applications, the longer it has to travel, the more room there is for error, right? And when there's error in genome coding, which takes place in the diameter of the nucleobase, you can have more issues. So a person is more likely to have issues by 0.3. Now on this one, you see it's 2.0 nanometers, but look at this one, 1.8. Now each one of those matter dramatically because not only are they closer, so the information travels faster, they also have double the base pairs. So you got to understand why they was like treating left-handed people like they treat them, and why they're looked at as different and treated as weird and stuff like that, because they have intricate significance to their cells. There's more that you can look up here, glycosal angle and all of that, which this is basically how you bond. So there's something called a glycosic bond, which is how you get the nucleobases to stick together. We about to get into that, but just keep in mind that this where it's at with these zDNA. They have more too. As y'all see, there's A, B, C. Now cDNA, just so that you know, this stands for complementary DNA, right? And it's not, let's see what they say. Hold on. cDNA. It is one of many possible double helical conformations of DNAs. Yeah, see, it's a conformation, like it conforms to it. Basically, cDNA just refers to like complementary DNA. It's what is produced after the RNA does its thing, right? Let me go into it real quick. Hold on. Yeah. Actually, it's a little bit different. Never mind. I'm confusing it with something else called cDNA. So there's something else called cDNA, which is it's a component of RNA. There's so many of these NAs, y'all. Like, we're going to go through them. Let's hurry up though, because I don't want to rush it, but you know what I'm saying? Listen, for everybody in here, sending positive vibes to everybody in here. I'm trying to not rush it because I want people to get their information, but I also know how people's like, their attention span is so short, like especially when you're not talking about something that's like super exciting to them. So, but this is extremely important information. What's up, 9IM? I send positive vibes to you and everybody else who in there. I love y'all. All right, let's keep going. So let's get back, right? Every one of these, y'all gotta keep in mind, like I said, you can do a whole lecture on every one of these links, but we're gonna go back. So the link that I had went into was double helix. So to reread this sentence, they basically was just telling you that the DNA is molecules, right? Which is a group of atoms composed of polynucleotide chains, right? Polynucleotides are made up of nucleosides, which are made up of 4 bases that we're about to go into once we get deeper in here. All right. So let's keep going so we can get down to it. All right. Now, DNA and ribonucleic acid are nucleic acids. Wait, why they make that sentence so short? A nucleic acids. Okay, so they just basically said DNA and RNA are nucleic acids. Duh. Alongside proteins, lipids, and complex carbohydrates, polysaccharides, which is basically sugar, right? And we're gonna do a whole thing on this. Hold on. Polysaccharides, nucleic acids are one of the 4 major types of macromolecules that are essential for all known forms of life. So let's go back real quick. Now, macromolecules, y'all remember what molecules are, right? Which is just a group of atoms, right? We all know the difference between micro and macro. If you don't, micro means really, really small. Macro means large. So based on these alone, you know what this is, but let's see what they say. Macromolecule is a very large molecule, such as protein, right? They always refer to proteins as macromolecules, which are basically a bunch of amino acids. And we'll go into that too, but they are composed of thousands of covalently bonded atoms. Covalent just refers to the electrons, the electrons that are in the atoms, their ability to bond to other atoms. So like when they say covalently, they just mean basically like it was done the right way. Covalently means right. You know what I'm saying? Because there's other ways to bond atoms and force it, and other kind of ways that they go about doing stuff. But this is the normal way. All right. Many macromolecules are polymers. Remember we talked about what polymers was. Polymers are just basically a group of molecules that can be chained together. So when you say polymer, you're basically saying a bunch of different atoms or a bunch of different molecules that can be chained together. So many macromolecules are polymers of smaller molecules called monomers. And remember, monomers, this is that shit where I was talking about. You go inside the building and go into the door, and then you go into the room, and then you go into the closet. You know what I'm saying? For anybody who's asking to come in when I'm doing lectures like this, I don't invite people in just because like I got a ton of information I'm trying to present, and it's not really meant to be more of a conversation. But anybody who want to like hop in after this, I can definitely go live. I just did a live for like 3 hours last night. Anybody was welcome to come in, and you always welcome to come in my lives. But when I'm doing something like this, I gotta, you know what I'm saying? I can't stop and have conversations. So my apologies. One love to y'all, the reflections. All right, my bad. I got sidetracked. All right, look. So just know now, when they say proteins, let me just tell y'all something real quick about proteins, right? So proteins, right, which is just some word that they make up, right? Proteins are large biomolecules, right? We know what this means based on this, right? We know what a molecule is. Molecule is a group of atoms. And I'm only going over this again just because the more you see it, the more it'll just be like night and day when you look at it, like, oh yeah. So bio refers to living organisms or living things. Molecules refers to groups of atoms. So they're basically saying proteins are large living groups of atoms, and macromolecules, right, which are just big groups of atoms that comprise one or more long chains of amino acid residues, right? Proteins perform a vast array of functions within organisms, including catalyzing metabolic reactions, DNA replication, responding to stimuli, blah, 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 right? What this means basically when they say, you know, catalyzing metabolic reactions. Metabolic refers to metabolism when your body is processing something. So for instance, let's use meat because people love to eat meat, which is killing you and giving you cancer. I'm sorry that you don't want to hear it, but that's what happens. But anyway, so when you're processing something, so when your body is metabolically processing meat, right? It has something called enzymes. And this catalyst, the proteins act as a catalyst to basically tell your RNA, we're going to get into that too, tell your RNA how to get information from what you're eating. This is why proteins are really important. But protein is just a word. That's why I say fuck they words. Who cares about their word? Would it just know that you get something from food when you eat it that gives instructions to your cells on how to react and how to do things, right? So that's what proteins are. They're macromolecules, but lipids, right? Lipids are just like layers. They're normally, let's see what they say. Hold on. In biology and biochemistry, a lipid is a macrobiomolecule that is soluble in non-polar solvents. Non-polar solvents are typically hydrocarbons used to dissolve other naturally occurring hydrocarbon lipid molecules, right? So basically what they're saying is lipids are capable of, like, they normally, like, you'll hear of lipids when you hear something called like a lipid bilayer, right? Lipids are normally like skin, like coats. You know what I'm saying? What they do is, and this is why they're insoluble in certain solvents, because they have to be able to administer transfers of information. Like for instance, there's a lipid bilayer around the nucleus envelope or the nuclei envelope of DNA cells. Not in us, but in eukaryotes. But that acts as sort of like an intermediary. So that's what they mean when they say lipids. Like basically just think layers. Like, you know what I'm saying? Yeah, they just make this stuff. All right. And complex carbohydrates, right? Hold on, what did they say? DNA and RNA are nucleic acids alongside protein, lipids, and complex carbohydrates, which is basically inorganic substances, but we'll get into that. Like polysaccharides, right? Nucleic acids are one of the four major macromolecules. All right, I read that. So polysaccharides, right? Let's go into this now. Let me see. Do they go into it later? I think we go into it later, but you know what? Let's go into it now because we here now. So let's talk about this. Let's talk about what carbohydrates are, right? Let's talk about what carbs are. Peace reflection. Shout out to everybody who in the comments. I seen Hades Patricky came in, Wave X now. I send positive vibes to anybody who come into the live and anybody who has the testicular fortitude to stay in. I love y'all. All right, now when we talk about carbohydrates, right? Hold on. Let me see something, right? So when we talk about carbohydrates, right? So carbs are sugar, right? This is what they're gonna tell you. Carbs are sugar. Let's look at a carbohydrate chart though. They should have some kind of chart that show you, right? Let's see. Matter of fact, let's do carbon chart. Hold up, y'all. Because you got to play around with these words a little bit, because in chemistry, words don't mean the same thing that they mean in other. Alright, so monosaturated. See what I'm saying? Monosaturates, we got to use that word. But they're carbohydrates, right? So let me look at saccharide chart, right? I should have already had this up, y'all. My apologies. I just be like because I want to show you the sugars, right? All right, here we go. All right. See? So look, where's a good one, right? So here's a good one. Damn, I think I done downloaded it. My bad, y'all. Come on, you sure big? Why y'all playing around? You really just gonna play around like that? You gonna play around? Alright, I'm gonna show you this. Look. So, these are carbohydrates are sugar. That's all it is. It refers to carbon, hydrogen, and oxygen. CHO, right? If you look up the chemical, let's look up real quick. Hold on. Chemical compound of carbon. We can do carbon or carbohydrates. Let's do carbon. I think we should actually do carbon. Let's do carbon. Y'all be playing with me, man. Stop playing with me. Will you show people the chemical compound? Hold on. What do they call it? Sometimes they call it the empirical compound, right? Empirical formula. They play with these words. CO2, that's carbon dioxide, you moron. All right, listen. Just listen to me when I tell y'all, okay? It's CHO. It's CHO, right? Stands for carbon, hydrogen, and oxygen, right? So carbohydrates is basically made up of sugar. Monosaccharides, this is real sugar, right? This is real sugar. So glucose, fructose, and galactose, right? Fructose is the more popular one. Well, actually, glucose is kind of popular, but fructose is more like readily available because fructose is found in fruit. That's like the main place that you'll find fructose. Now, these other ones, first of all, you already know this one ain't shit. But these other ones, these are all, especially this is a killer right here, starch. But these are all where they take a bunch of these molecules and they bond them together through a process called isomerization or polymerization. And there's other words and terms that they use as well because they got a different word. Like I say, fuck their words, but they got a different word based on every chemical change that takes place, right? And if you look up these things, right, if you try to look up where do you find glucose, I bet you they're going to tell you to look. Where can glucose be found, right? And this is necessary, y'all, because we connecting dots here too. All right, so glucose belongs to the family of carbohydrates. It is a monosaccharide, simple sugar, naturally present in all living beings on Earth and in their most important source of energy. It is found in high quantities in fruit, right? Vegetables and honey, right? So when we look up, let's see glucose, let's see fructose, right? Fructose. Found naturally in fruits, fruit juices, vegetables, and honey, right? Now, let's do galactose as well because I like galactose because I like that it got that galact in the front. It make me, I'm a big fan of Galactus from Marvel. All right, it is found in nature, right? This is how you know these are real, right? Found in nature with other sugars, right? Now, see how they try to combine it and run it together? It is usually found in nature combined with other sugars, as for example, in lactose. Now, the reason they saying that is because lactose is a combination of galactose and glucose mixed together. So when you take those two and you mix them together, you get lactose, which is why they trying to say combine with other like, come on, man, but that's an inorganic substance. But let's say lactose. Now, if you notice all of those ones that we looked up, they said they came from nature. They can be found in plants, all of the things that the sun gives support to. Now, look when we look at lactose. Lactose is a sugar found primarily in milk and other dairy products. You know what I'm saying? Enzyme called lactase. right? All of this is foreign. All of this is foreign. And I'm showing you this just so that, like, we could do the same thing with sucrose. And there's one other thing I want to show you too before we go back. Where can sucrose be found, right? Let's see what they say about sucrose. Sugar cane, which is a hybrid plant that's not real. Cane is not real. Sugar beets, sugar maple sap, dates, honey. Honey is probably the only thing on here that is a natural thing. But you know what I'm saying? Like I said, a lot of time they're going to send you to something that is inorganic when you're talking about an inorganic source. And sucrose is inorganic. It is something that is made in a lab through a process. Same thing with maltose, right? Let's look up maltose. And we about to move on, y'all. I apologize for y'all that's like, why you keep talking about sugar? Leave it alone. But I want you to see the ideology and the concept of what I'm saying behind this. Shout out to It's Just in Phase, sending positive vibes to you, Reflection. One love to everybody who came in. Maltose, or malt sugar, is an intermediate in the intestinal digestion of glycogen and starch, and is found in germinating grains and other plants and vegetables, right? Now, germinating grains, they're talking about grains that they made. This is not natural. These are not natural, and neither is starch. Starch is definitely not natural, although it does occur naturally in plants during photosynthesis and different kinds of processes that plants have. That they're talking about is not natural. So I just wanted people, when you see carbohydrates, because you always going to hear, eat your carbs, you need your carbs, to understand that these are what carbohydrates are. So when you look on the back of your food and you see disaccharides, that's basically carbs that are di, meaning split. Di in chemistry means duality, double, right? So that's why they say two sugar. Ali just refers to few, like a couple. They normally use it for different amounts, but this one says 2 to 10. So it's just basically in between this one and this one. And poly means 10 or more. And if you look these up, right, this is one thing I wanted to show y'all, right? What is let me show you something real quick, right? I'm gonna type in this, right? What is the chemical compound of an apple, right? Because they say what? An apple a day. Oh, that's because I'm on images, y'all. That's why it wasn't popping up all that time. Because I was like, yo, why they playing with me, not pulling it up? Why they not? You know, this is so crazy because, like, when I first was learning this, I had all of it pop up. Chemical composition of an apple. They playing. Alright. Just know that it's chill. Now, this is a specific acid that they're showing you, but there's one that refers to an apple itself. And it's just chill. That's all. That's all it is. They doing a whole bunch of crap here. But just know that it's chill. It refers to carbon, hydrogen, and oxygen. That's the only thing that you need. And this is not the exact atomic numbers, 4, 6, and 5 associated with an apple, but this is very close. It's like 7, 12, and 6 or something like that, right? For an apple. Now, if you type in, what is the chemical composition of sucrose? Look at this. C12, H22, O11, right? Now you got to understand what this does to your insides when you eat this, because you know the phrase, what is it? An apple a day keeps the doctor away. You know what I'm saying? When you look at an apple and see that an apple has like C7, H, 12, O, whatever, this is more than double in some aspects of what that is. And mind you, this is just one. They bond these together in polyforms. And this is not even this is table sugar. right? Sucrose is a type of sugar made up of one molecule of glucose and one molecule of fructose, right? Anytime they say made up of one and one, that's GMO, bro. That's genetic modification. So table sugar is genetically modified. And they just told you. You know what I'm saying? Like, that's what it is. So, and it causes health issues. Like, it really causes health issues. We don't have to go into that, though. This is about DNA. Let me get back to what I was doing. All right, my apologies, y'all. All right, so that's what I just wanted y'all to know about polysaccharides, is that basically, I'm gonna just leave that alone. Yeah, just know that when they say carbohydrates, they're talking about sugar, and this is what polysaccharides are. Because when we think sugar, I just feel like I've always felt like sugar was like just this sweet thing that you eat. Like, oh, sugar is just flavoring or seasoning, you know. But sugar is actually a chemical structure that does a lot more than taste good. In fact, a lot of sugars don't even have a taste. They're odorless and tasteless, but they add extra things to them to make them have taste. Other than, you know, the natural ones, glucose, fructose, and galactose. All right, but look, let's move forward. So the two DNA strands are known as polynucleotides, right? As they are composed of simpler monomeric units called nucleotides, right? So polynucleotides, again, is just a group of nucleotides, and nucleotides are a group of nucleobases that make up the base structure of what these DNA backbones are, right? So each nucleotide is composed of one of four nitrogen containing nucleobases. That's cytosine, guanine, adenine, and thymine. There is another one called uracil. We'll go over that in a second because uracil is specific to RNA and there's a purpose that it has. But I just want to go through these so that y'all can know. When you're looking at these so it don't look so strange, right? When you're looking at all of these dots, right? Or when you're looking at any of these structures, right? This is what these are. So when you're looking at all of these, adenine, thymine, it's always going to be ATGC because GC works together. So these two, let's scroll back up. My bad, John. I'm going everywhere. So cytosine and guanine work together. And we're going to learn that in 1 second. And adenine and thymine work together. We're going to go over what these are real quick. Peace to you, Reflection. Shout out to everybody in the comments, sending nothing but love to y'all. Watch it back if you get a chance so that you can see everything, all right? But, all right, so each nucleotide is composed of one of four nitrogen-containing nucleobases, right? Now, for those who wonder, like, why is it containing nitrogen, right? There's something called a nitrogen cycle, right? Let's see what they say. Hold on. I'm going to see how quick I can try to explain this. Nucleobases are known as nitrogenous bases, or often simply bases, are nitrogen-containing biological compounds that form nucleosides, right? Now, the reason that you want to know this is there's something called a nitrogen cycle, right? Which just refers to nitrogen's ability to, hold on. We're going to come across it at another time. Just know that there's a reason that nitrogen is here. There's a reason that so let's go into this. This might take us into it. So let's go into nucleotides because they're going to talk about phosphate, which is why you need. All right. So what is a nucleotide? Nucleotide is organic molecules. We know molecules are what? Groups of atoms consisting of a nucleoside, right? This is that shit where I said you got a house and then you got a door in the house and then you got a closet in that door and then you got a trunk inside that closet, right? So this is the room door, right? This is the closet in the room. Well, nucleosides are the closet. This is the closet in the room, right? And let's get to, this is the trunk inside the closet, right? Glycosamines, and fuck they words, okay? Glyco just refers to sugar and amines refers to amino acids. This is basically them just saying that they bond amino acids, and amino acids come from proteins. Proteins are a group of amino acids, right? So nucleosides are just bonded aminos that can be thought of as nucleotides without a phosphate. Now, This is where nucleosides become nucleotides when you have a phosphate. And the reason you need a phosphate is because, well, let's see what they say. In chemistry, a phosphate is an anion, salt or functional group or ester derived from a phosphoric acid. It basically has the ability to remove hydrogen, which is why you need it, right? You see how H3 is here for phosphate, but then when you look over here, it's H2, and this is the chemical compound of adenosine, and adenosine is a nucleoside or nucleotide. This is a nucleotide, right? So I don't want to confuse people. I apologize. I'm really just kind of going over it real quick. So let's go back to nucleotides. So nucleotides are organic molecules consisting of a nucleoside and a phosphate. They serve as monomeric units of the nucleic acid, right? Polymers, DNA and RNA. So basically, nucleotides are part of DNA and RNA, which are essential biomolecules within all life forms, right? This was an unnecessary statement. Like they're just saying the same shit over and over again. Hold on. See if we can find something that actually has some substance to it, because they just like they just have these sentences with a bunch of big words that sound all cool. Like, this is what genetic coding looks like, by the way, so that y'all see. We're going to get into that in a minute. Genome coding. So when you see letters like UUTG, GGUTA, this is referring to these uracil. Uracil is, remember we said there's 4 bases, right? Adenine, cytosine, guanine, and thymine. Uracil is a base. Uracil is thymine, but in RNA, right? So in RNA, uracil converts to thymine, and we're about to go over that. Maybe I'm spending too much time on this. Let's go back. All right, let's just go down. All right, so each nucleotide is composed of one of 4 nitrogen-containing nucleobases, which is cytosine, guanine, adenine, and thymine, right? Which we just went over. A sugar called deoxyribose. Now, this is what I was telling you about deoxyribonucleic acid. Deoxy refers to the removal of an oxygen atom. Ribo refers to ribose. And nucleic acid refers to the base acid that is there, right? So deoxyribose, let's see what they say about this, if we can learn anything more. It's basically just showing you the formula, which is CHO, like I told you, it's CHO, but they have different variations of it, right? When you remove oxygen from it, you can put it in different ways. So without causing too much confusion, just know that it's derived. So deoxy sugar or deoxyribose is derived from the sugar ribose by loss of an oxygen atom. And this is why you need phosphate, because it removes oxygen. Peace reflection. So just, we don't even need to go into all of that. Just know these parts so that when you hear this stuff, you're like, oh, okay, it's a phosphate group. Now, the nucleotides are joined to one another in a chain by covalent bonds known as phosphodiester linkage between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar phosphate backbone, right? And they basically just talking about this, letting you know how this is structured, right? Not really all that important. The nitrogenous bases of the 2 separate polynucleotide strands are bound together according to base pairing. Now, this is what I was telling y'all earlier that we was going to get into, base pairing. Base pairing, so you see this right here? This is a zoom in to this, right? So when you see these boxes, these are base pairs. When you see that connected like that, this is a like a microscopic view of what this is. So these are the base pairs, like right there where you have those 2 joining. That's called a base pair, just so that you know. All right, and the base pairs have rules. A with T, C with G. Remember I told you C and G work together. As long as you remember that, then you can remember the other 2, and vice versa. But that's why you got T to A, right? Thymine to adenine, right? Adenine is responsible. We're about to go into each one of these. Adenine is responsible for providing power and energy. C and G just kind of help with transport and stuff like that, right? So we also going to discuss like what things you can eat to give you these things too, because if you're trying to like superpower your DNA, you got to know what's in it. You got to know how it works and what it does. Like, you know what I'm saying? So the nitrogenous bases of the 2 separate polynucleotide strands are bound together according to base pairing rules, right? A with T and C with G. with hydrogen bonds, right, to make double-stranded DNA. And these are the hydrogen bonds that they're talking about. These links right here. These is hydrogen in there. See the little dot for hydrogen? There's a little dot there. These are the hydrogen bonds. That's linking it together. Just know that. So it doesn't seem too weird. Peace to everybody who came into the live and anybody who's still in here. One love to you. All right, the complementary nitrogenous bases are divided into two groups: pyrimidines and purines. In DNA, the pyrimidines and thymine and cytosine, and the purines are adenine and guanine. Now, this can be a little bit confusing. Sometimes all you need to know about these two things, because you'll look at this and be like, yo, what does this mean? So what is pyrimidines? These are basically like a structure. Like, imagine if I was to say we building a house. This house is going to be a square and this house is going to be a triangle, right? If we were saying that, it would sound a lot easier than this, right? But just know that. Now, we can go into it so you can see what they say, because you always want to see what they say. Pyrimidine is an aromatic heterocyclic organic compound. See how they make this? You see that? You would look at that and be like, what, nigga? Is that a bike that's gay or a bike that's straight? I don't know what that is. But look. Pyrimidine is an aromatic heterocyclic organic compound similar to pyridine, right? One of the three diazines. It has the nitrogen atoms at position 1 and 3 in the ring, right? So just know this, right? This is what they're just saying, right? 1 and 3, right? They're just saying a pyrimidine is a bond that's going to have nitrogen atoms here and here. That's all they're saying. You know what I'm saying? But when you throw it in there, the pyrimidines, they just basically saying when you see a nitrogen atom at 1 and 3 on a 6-point, bond, chemical bond, what they call a heterocyclic, right? This is called heterocyclic. My bad, I should have mentioned that. When they say heterocyclic, right? Let's see what they say. Hold on. My bad, y'all. Heterocyclic compound is a cyclic compound that has atoms of at least 2 different elements, right? That's hence your hetero, right? So this is heterocyclic. If it was homocyclic, it would be nitrogen atoms on each one. But because it's heterocyclic, it only has nitrogen atoms at 1 and 3. That's basically all you need to know. That's what pyrimidine is. Because the way they talk about it, they make it out like pyrimidine is its own chemical or something. Like you would think, you know what I'm saying? When you look these things up, and then it says it's similar to pyridine, right? What is pyridine? My bad. Hold on. Pyridine is a basic heterocyclic with the chemical formula CHN. Right. So, you know what I'm saying? And these are diazines, which are what? Probably just groups of structures. Diazines are a group of organic compounds that having the molecular formula CHN. Right. So yeah, basically they're just saying anytime that you have a group of things that compose of carbon, hydrogen, and nitrogen, they can be ordered in different fashions. This is pyridine. This is the type. What's up, Chanel? I appreciate you for coming into the live. You just missed like me talking about some stuff rudimentarily about DNA. I'm just going over DNA, putting it into layman's terms because they try to make this shit complicated when it's really not that serious. Well, it is that serious, but you just, Anyway, all right, so pyrimidine just refers to, again, nitrogen being at 1 and 3, right? And then when we go to purine is a little bit more basic, right? And for those who may have missed it, these are Neutrogene bases. These are what we call nitrogenous bases, right? These are what your DNA is made out of, right? Because your DNA, deoxyribonucleic acid, is made up of polynucleotide chains, right? These polynucleotide chains are composed of structures. They're composed of bases, which are guanine. We just talked about these: cytosine, guanine, adenine, and thymine, right? But those bases are structured in a certain format, and that's what these are. So purines is how the connection between C and G. Let's go take a look at it. Let's see what they say first. My bad. Purine is a heterocyclic aromatic organic compound, and we know heterocyclic just means what? It's a circular shape that has multiple different elements. That's what they mean by that, consisting of 2 rings. So basically purine is basically pyrimidine, but with 2 rings. So see? Now, let's see if they tell you what those points are. Hold on. Yeah, so All right, so it looks like based on reading this, like it basically looks like this changes depending on what the process, what the coding is that's taking place is, because you know there's genomes and coding that takes place during your RNA being processed, which we're about to get into actually in a second too. All right, so that was that. Now I forgot what I just went into to show y'all that. Oh, that was pyrimidines. But let's go into each one of these real quick, right? Each one of these nucleobases, right? So that you can know something about what these are. Kelbocyrus sending positive, or Kelbiocyrus sending positive vibes to you, reflection. I appreciate you for coming into the live. One love to everybody who has the testicular fortitude to stick around for stuff like this when it's not entertaining, right? All right, so cytosine, right? The first one. I'm just going to show y'all something real quick. Now, all of these, remember, you got a house, and then inside the house, you got a room, and then inside the room, you got a closet, right? Cytosine or nucleobase is the house. Cytosine is the room, right? So let's go into the room. Now, in the room, we got a closet, right? Matter of fact, let me do it a different way. Hold on. Let me do it a different way. In the room, we got a closet, right? And that closet is this, right? Because cytosine is just a result of this. So this is really what you want to know. Or cytosine, right? This is cytidine, right? Now cytidine is a nucleoside, and remember, nucleosides are simply, they just haven't come into contact with a nitrogen phosphate yet, right? And you got to remember, a phosphate removes hydrogen, right? Which will break that off there. That's how that all works together. It's not really necessary that you know all of that, but just understand, like, to a conceptual level, what they're talking about. But cytidine is a nucleoside molecule that is formed when cytosine is attached to a ribose ring, right? So basically, the nucleoside of cytosine is cytidine. Right. Cytidine is a nucleoside molecule. See, when they be confusing me because they say nucleoside and nucleotide. Nucleoside is a molecules within nucleotide. Nucleoside molecule that is formed when cytosine is attached to a ribose ring. Right. So basically just know that this chemical is necessary in order to make this. Right. So if you wanted to try that, so you could look up, like, right, let's see. Right? I was looking up maltose earlier, which I explained to y'all is a fake sugar that you do not want to put in your body. That's why most people are sick. It's crazy all of these people so worried about PC reflection. What's up, Bugsy? All these people so worried about this fake virus that they got out and all of this mad stuff, but you got a cart full of a bunch of stuff that's killing you, and you don't even want to know about it. It's so crazy. Like, people be in the market. Oh yeah, cytidine. People be in the market talking about, oh, can you put your mask on? Oh my goodness, put your mask on, while they walking around with a cart full of death. They literally walking around with a cart that's going to give them lysine, pyrrolysine, all these types of, bacteria that's active. Anyway, it just annoys me, man, because people can be so ignorant. Like, how can you be so ignorant that you care to protect yourself from this, but you got a cart full of stuff that's going to kill you? Like, and you don't even want to know. If I start talking to you about how cheese is going to kill you, you going to tell me get out your face. But then when they turn on the TV, you know what I'm saying, and start telling you about this fake virus thing. All right, anyway. All right, so look, cytidine. So look what they tell you. You see the first thing they do? They always point you to meat. Organ meats, right? And the reason they telling you organ meats, because organ meats are going to have enzymes in them, right? Proteins, macromolecules. And the purpose of these are going to be to instruct your DNA and your RNA, right? So they telling you that you can get cytosine from foods with look, I didn't even see that, with high RNA. Yeah, because there's RNA in organ meat, because that's how the cells know how to do what they do in the organ. And then they tell you in yeast. Brewer's yeast, bro, from beer. That's the best you can come up with to tell me to get this chemical. And this is the chemical that's the base in your DNA, right? Now let's do this, right? Let's look up what vegetables. Watch this, right? This is how it's worth $1,000 and nowhere to look, but it's worth $1 million to know what to look for. What vegetables can cytidine be found in, right? Because they ain't say shit about vegetables, did they? Hold on. See, they always trying. Hold on, y'all. Give me 2 seconds. Hold on. Give me 1 second. Cause they be real. They be. Because right, here's what happens. Right, they got it real. They got it real slick for you. You know what I mean? They got it real slick. Like they'll tell you, oh, find it here and here. But I found something that said that cytosine was actually found in specific plants, and it was actually quite rich in plants. But they won't tell you that, and they won't show you that, because if you ate plants, then you won't die as fast, and you won't have to go back to the doctor and have them give you a bunch of prescriptions that's killing you slowly. Probiotics, which are basically GMO chemicals that they put in you to kill you off, and basically instruct your cells on how to get sick in the future, or how to not deal with other things that they're going to come out with later, which are building blocks. This is why they keep a record of everything you take. Hold on, y'all. Alright, so cytosine. Listen, hold on. What? Where can cytosine or cytidine be found? If I don't find this, I'm gonna just move forward, y'all, because I don't wanna waste too much time with this. I just wanna show y'all. All right, so peep this. I'm gonna be honest with you. I can't find it right now. I'm pretty sure I will find it if I kept looking. But I found something that was explaining. Let's get off of this. I found something that was explaining that you can find cytidine in plants, and same thing with guanine. Like guanine, right? When we go to guanine. Matter of fact, hold on. Was it down here? Hold on. Because sometimes they'll have Let me go to guanine real quick. Guanine was the next one that we was about to go to, y'all. So let's just go into it while we here and get it over with so we can move forward. I basically just wanted y'all to know what they all do. Yeah. See, this is telling you a little bit about cloning right here. We're going to get into this too, but look at this. When we go to guanine under synthesis, they say trace amounts of guanine formed by the polymerization of ammonium cyanide, right? 2 experiments conducted by Levy, whatever his name is, showed that heating, blah, 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 blah. So what this tells you is that they playing around with DNA strands through polymerization, which we know what polymers are, right? It's a group of molecules that are, like a bunch of molecules that are together. And ation just refers to a process that they're doing, that they're putting them through. So they found out a way to do something with that. We're going to go more into depth in that. Listen, I'm not going to focus too much on this, but just know that each one of these adenine, cytosine, and thymine, and uracil, and guanine, these all come from plants. They can come from meat too, but there's other stuff that comes with the meat too that is negative for you. And they're not going to tell you about that. You know what I'm saying? They just tell you about the, so just know that the purpose of cytosine is to act alongside, hold on. I just wanted to cover them real quick, each, individually. Yeah, see, so it talks about chemical reactions, right? Cytosine can be found as part of DNA, as part of RNA, or as a nucleotide. It can act as a cofactor to enzymes and can transfer a phosphate to convert adenosine diphosphate, ADP, to triphosphate, right? So this basically just tells you cytosine is meant to help adenosine, which comes from adenine. When it's mixed with a phosphate, it comes adenine. Remember, that's the room in the closet, right? Adenosine is the room. Adenine is the closet, right? But you get the room when you put everything together with phosphate in the closet, right? So just know that cytosine helps out adenine, right? Guanine helps out cytosine, but let's see if we can find something real quick that will show us. So we about to get past this, y'all. My bad. I know it's taking a minute. I just, I don't have this all written out enough, and I just kind of go over what I learned, and You know what I'm saying? Let's see what they say about with the formula CHO. It's always Chino. It's always Cho. And you know what's crazy? This is, look at this. Let me show y'all something about this because I actually happen to notice. Chino. Watch this. Let me show you something. Hold on. Give me 2 seconds. Give me 2 seconds real quick. Watch this. Interesting, right? What is the chemical formula of... Alright. Peep this. Y'all know what serotonin is? Now, you know what serotonin is? Serotonin is a chemical that makes you happy. That's essentially what it is. It's a neurotransmitter, right? Modulating mood, condition, reward, learning, memory, numerous physiological processes. This can be created with this. CHNO? CHNO, carbon, hydrogen, nitrogen, and oxygen, right? So through a process called polymerization, do you now understand how you can be made happy with food you eat? I can take a bunch of rocks and put them together and put this chemical makeup in them, and you can eat rocks and feel happy. This is a chemical reaction that takes place in your body. Now, when we go back to guanine, look at this. This is the formula for guanine. Now, granted, the atomic numbers are different. This one is 555 for guanine, and for serotonin it's 1012-2. You know what I mean? You need less nitrogen. But you got to understand that when you understand how to do isomerization, polymerization, hydroxyl bonding, all these kind of things, you can figure out what atoms and chemicals you need to put together to make this 555. It's not hard. You know what I'm saying? So you could literally, not 555, that would make it guanine, but make different sugars, right? Which guanine is. Well, it's not a sugar, but it's a ribose. Ribose is a sugar, which ribose is made up of the nucleobase. These all form ribose. You know what I'm saying? This is why it's deoxyribose nucleic acid. Like, all of these together come together to form ribose, which we're about to get into. Ribose is just a sugar. All right. So just know that guanine helps with this. But let me see if it just says specifically. Guanine can be hydrolyzed, right, with strong acid to glycine. And this is all witchcraft. This shit is not normal. Hydrolyzed means the removal of an oxygen atom or water. Hydrolysis is a chemical reaction in which a molecule of water breaks one or more chemical bonds. The term is used broadly in substitution and elimination. Listen, let me just say something for a second, right? Right? What is the reason? Because I always have to point out the diabolicalness in this shit, right? Imagine if you needed me to pick your daughter up from school and take her home, right? And all you know is that I'm a driver. I pick your daughter up, I take her home. You saw my history. You spoke to people who work with me. You know that I got other clients. I'm good, right? So all I got to do. What reason would you need to know what size the tires on my car is? What the air pressure in each tire is? What type of oil comes out the back of the car? What type of compartments that I have? Why would you need to know all of that? You understand? Unless you have, all you need to know is that I'm picking your daughter up and I'm taking her home and I have a business that I do. Why do you need to know all of this stuff about my car unless you plan to fuck with it? You know what I'm saying? Unless you plan to flatten my tire, or unless you plan to try to change out my tire or switch my car. You understand? So when you apply that concept to what I'm saying, why do you need to know this? You got to understand, they put years and years of teams of study together to learn this stuff about you. They not learning this stuff because they want to shake hands and be friends. You understand? That's never been, history. You don't know anything about history if you think that. Anyway, let's move forward, y'all. My bad. I just gotta shout out to everybody in the comments. I see the TYM just came in. Guapo Chris, or Guap Chris, sending positive vibes to you, Reflection. I appreciate you for coming in. I always appreciate everybody who come into my lives. You know what I'm saying? Because it matters to me that you're here. You could be doing anything. You know what I'm saying? So it means a lot to me. So let's jump back on track here. I forgot where I was, y'all, because adenine, let's go to adenine. Adenine is one of the more important ones. I mean, they're all important, but I think this is the most important, but this one is pretty important. So adenine, right, which is adenine is the room, right? And adenosine is the closet. So let's go in. What is adenine, right? You have nuclear bases, right? So adenine is a nucleobase, right? And a nucleobase is just basically those groups of, it's one of those four that make the nucleoside, right? A nucleobase is a component of the nucleoside, right? If that's making any sense. If it don't to anybody, please let me know and I'll cover it again, but just know that adenine is a nucleobase. It is one of the four nucleobases in the nucleic acid of DNA that are represented by the letters G, guanine, right? C, cytosine, right? A, adenine, and T, thymine, right? We also have uracil. We'll get into that, but keep in mind, uracil is only relative to RNA, not DNA, because uracil converts into thymine when it comes to DNA. But the three others are guanine, cytosine, and thymine. Its derivatives have a variety of roles in biochemistry, including cellular respiration, right? So let's just go to this so you can see what it does, right? Because reflection, and when I say reflection, I'm talking about collide, right? What's up, Roadblock D? Sending positive vibes to you, reflection. I appreciate you joining us. Reflection, talking about collide, he was talking about how adenosine is responsible for your neurons, right? And adenosine is one of the things that throws off your imbalance in your neurons. Now let's take a look at what adenine does, because adenine, again, is a form of adenosine, right? So adenine is one of the four, blah, 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 right? The three, blah, blah. Its derivatives have a variety of roles in biochemistry, including cellular respiration, right, which just means to heal the cells as you go along and provide it with oxygen and such, transporting information. In the form of both the energy-rich adenosine phosphate, triphosphate, ATP, and the cofactors, nicotinamide, adenine, dinucleotide. Boy, they be killing me with these words. Flavin, whatever, we're not going to say all those, and coenzyme, right? Let me see what these is real quick, because I actually didn't look into that. Hold on. NAD is a coenzyme central to metabolism found in all living cells. See, there's so many different levels to this. Like, you know what I'm saying? Every time there's a process, so like even in RNA, when you're transferring information, most people know about something called mRNA, right? Which is messenger RNA. But there's processes underneath messenger RNA. Like there's so much. All right, let's move on. Right, the shape of adenine is complementary to either thymine in DNA or uracil in RNA, right? And the reason they saying that is because adenine feeds directly from thymine. Remember, base pairing. You remember base pairing rules, right? Adenine feeds directly from thymine, and thymine is converted from uracil. Uracil is specific to RNA though. Let's look into that so you can see what I mean. Uracil is one of the four nucleobases in the nucleic acid RNA that are represented by the letters A, G, C, U. That's adenine, guanine, cytosine, and uracil, right? Thymine is specific to DNA, not RNA. The others are adenine, cytosine, and guanine, right? In RNA, uracil binds to adenine via two hydrogen bonds. In DNA, the uracil nucleobase, let's go real quick. The uracil nucleobase is replaced by thymine, right? Uracil is a demethylated form of thymine. So that what that tells you is once they take the methyl out of it, then it becomes thymine, right? And demethylation is a chemical process resulting in the removal of a methyl group, right? And a methyl group is carbon and hydrogen 3, right? So when you take that out, then you now have thymine, and that's what's sent over to DNA. Shout out to everybody in the comments. One love to you, Reflections. I appreciate you for coming in, right? And then you get into, it's a little bit more, then you get into why nitrogen is involved and why phosphates are involved, because they assist as catalysts in the removal of hydrogen atoms, And it all just comes together, you know what I'm saying? But it's a lot more. I would just recommend y'all take a look into it. I don't want to get all into that. The main thing is just knowing what these things are. So just know that adenine is responsible for sending power, you know what I'm saying? And they say it's the shape of this because, again, based on the way the thymine comes in, is going to determine what adenine does. So you want to keep that in mind. Adenine works directly it works alongside thymine, right? Now, adenosine, right, which is, remember, adenine is the room, adenosine is the closet in the room, right? They always have these extra layers, right? Adenosine, and fuck they words, Adenosine is an organic compound that occurs widely in nature in the form of diverse derivatives. The molecule, which is a group of atoms, right? The molecule consists of an adenine attached to a ribose. Now, ribose is sugar through something called a glycosidic bond. This is how they make fake sugar. Like those disaccharates that they make, fructose and lactose and all that crap, right? But our body does it naturally. So just know that adenosine helps with that happening, and adenine is the result of that and communicates with the rest of the body. That's why it says its role is cellular respiration, right? Which, let's just see what that means real quick. Cellular respiration is a set of metabolic reactions and processes that take place in the cells of organisms to convert chemical energy, right? From oxygen, right? Like I was saying. So it's to move oxygen, and, this works hand in hand with thymine because thymine is connected to your T cells, right? This is why A and T work together. And your T cells, for those who don't know, shout out to everybody who came into the live. One love, Reflections. I appreciate y'all for being in here. For those who don't know, your T cells are directly tied to your thymine, right? And the difference between T cells and regular blood cells is that they come with like a specific script of what to look for. So they have like more information than the other cells. They call white blood cells. They have more information than the other blood cells on things. Let's go into thymine real quick though, because that's going to take us into it a little bit, right? Thymine. Thymine is one of the 4 nucleobases in the nucleic acid of DNA that are represented by the letters GCAT, right? Which is guanine, cytosine, adenine, and thymine, right? The other, and uracil, but uracil is associated with RNA, right? The others are adenine, guanine, and cytosine. Thymine is also known as 5-methyl-uracil. And that's because that's, the process of uracil when it converts to thymine is the removal of methyl. We just looked that up, right? We know. All right, a pyrimidine nucleobase, right? And that just refers to the 5-point thing that we showed. The pyrimidine is a structure. Think about pyramid. It's a structure. You know what I'm saying? It's just a structure where N is at 1 and 3, right? Remember? In RNA, thymine is replaced by the nucleobase, right? So they just reiterate, and let's see a little bit more though, so you can see. Now, thymine was first isolated in 1893, which is 993, looking at those numbers, but by whatever his name is, who cares? From calf thymus glands, right? So thymus, right? When we go into the thymus, this is what thymine is based on, right? The thymus is a specialized primary lymphoid organ of the immune system. Within the thymus cell lymphocytes, or T cells, right? T cells within the thymus. Wait, what? Was that an incomplete sentence? Within the thymus cell lymphocytes, or T cells, mature. Oh, I was missing that word, mature. T cells are critical to the adaptive immune system, where the body adapts specifically to foreign invaders, right? So this is how everything works together, and you can figure out how your body gets sick. Also, mucous membranes. I'm gonna have to go over mucous membranes as well, because again, mucous membranes coat your whole body, your respiratory system, your sexual, your anus, your penis, your vagina, your respiratory, your oral, your eyes. All of that is covered by your mucus, what they call your mucosa or your mucous membrane. which is tied into this. And it's actually the first step in between you getting sick because of something outside of you. That's why, like Dr. Sebi was teaching, that the only sickness there is the mucous membrane. Because in order for you to be sick, your mucous membrane has to be ruptured. That's the only way that can happen. One love to everybody in the comments too. Sending positive vibes to you. The thymus is located, and this is directly tied into the thymine. Remember, the thymus is located in the upper front part of the chest, right? So here, it's like in the middle of your sternum, right? In the anterior superior medius, fuck they names, man. This is the word that they got for this, all of this, for that here, like whatever. Behind the sternum. Oh, that's what I just said. Behind the sternum and in front of the heart. It is made up of two lobes, right? Two lobes, each consisting of central medulla and outer cortex surrounded by a capsule. The thymus is made up of immature T cells and thymocytes. What are thymocytes? Hold on real quick, y'all. My bad. This is my own knowledge now, because I ain't look into that. Thymocytes are produced... Wait. Thymocyte is an immune cell present in the thymus. Okay, so they're basically cells that are unlike the T cells. All right, so that's enough for this. We don't got to go into it. If you go down, you can see a little bit more about it, about how wide it is, how it interacts with other body parts, the trachea and things like that. And you can learn more about the blood and nerve supply, the arteries, how basically your T cells can be affected by things you eat. And this is why it would be important to know these things about yourself in order to like really try to figure out what is the issue with your body, right? I'm just looking at this real quick to make sure there's nothing that I wanted to like touch on. Whatever, let's go back. All right, that's the thymine. So just know the thymine is important because it receives power from the adenine, which we just went over how that happens, right? All right, so let's move forward. The nucleotides are joined. The nucleotides are joined to one another in a chain by covalent bonds known as the phosphodiester linkage. Blah blah blah. I think I read that already. Yeah, I might have read that already. We could go down past that, y'all, because we don't need to be doubling up. Now, both strands of double-stranded DNA store the same biological information. That's basically telling you that these two store the same biological information, right? This information is replicated when the two strands separate. A large part of DNA, more than 90% for humans, is non-coding, right? Meaning that these sections do not serve as patterns for protein sequences. Now, when they say protein sequences, you got to remember what proteins are. They're amino acids, right? And amino acids' whole job is basically to be broken down and they act as catalysts for your body to understand what you're doing, right? So when you eat something, your body breaks it down and it gets instructions from the amino acids. And you can look into this and see it in depth and everything, but I'm just trying to tell you what it is, all right? The two strands of DNA run in opposite direction to each other and are thus anti-parallel. Attached to each sugar is one of 4 types of nucleobases. They're the ones that we just went over, right? Let me just look over this and see if we need to. All right, so let's go into RNA, right? Attached to these 4 types of nucleobases, it is the sequence of these 4 nucleobases along the backbone that encodes genetic information. Now that's important for the simple fact that you got to understand how DNA is transferred and how you learn, because throughout the day you have millions of blood cells whose job is specifically to reteach you stuff, and they do this through something called coding, right? Which we'll go into coding. I think a little bit down we'll go into coding. We nowhere near done. There's a million things we can learn, but I'm almost done. We're gonna go into it right here. I'm gonna go into it right here. But for here, now it says RNA strands are created using DNA strands as a template in a process called transcription, right? Where DNA bases are exchanged for their corresponding bases, except in the case of thymine, for which RNA substitutes uracil, right? We know that. Under the genetic code, these RNA strands specify the sequence of amino acids within proteins in a process called translation, right? So let's dissect this a little bit. So transcription is actually a process within RNA. So I don't know if I want to go to this or the other one first, but let's go to RNA first, right? First off, I wanted y'all to see what ribose is, right? Because ribonucleic acid refers to ribose, which is a sugar that is, see how they say ribosomes and ribosomal RNA? There's a bunch of different RNA, bro. There's rRNA, there's mRNA, there's RNR. Like there's several levels to the way RNA works, because a lot of people think, oh, well, your mRNA tells you your DNA and that's it. Like, no, you have tRNA, which and it's not really necessary to know all of this, honestly, because fuck the words. All you need to know is that RNA basically is responsible for what roles? For what biological roles? Coding, decoding, regulation, and expression of genes, right? Gene expression. That's really what you need to know about RNA. I'm just showing you this so that if you want to go into many other ways if you want to go into the depth of how it works, you can do that, because sometimes it's very helpful to understand things like, what it's composed of. And like each one of these, you can look into these. These are the backbone, the acids, you know what I'm saying, that make, or the nucleobases that make the acid. You can look into what you can eat to give you these, and vegetables in particular. And this will help you, you know what I'm saying? This will help you in terms of your body's ability to accept photon energy as well, which is what comes from the sun. This is where this awakening that they're talking about us having and comes from. But you have to have your DNA in a position where it's able to operate the way it should. Like, you know what I'm saying? That's what it's about right now with this jab. But anyway, I want to also touch on, well, I'm not going to mix that into this. Just know that this is what RNA is. There's several different levels of RNA. I'm not going to go into all of it, but certain processes require different things. Like each one of these, if we went into these and we went into coding, right? All right, you know what? Let's do it. Whatever. We don't got to make it long, but go into coding, right? This is what I was telling you about the codes. So I got hair on my hand. GCU, right? That's guanine, cytosine, uracil, right? This is a RNA codon. And first of all, you know that because it's U, and U is only affiliated with RNA. Once it's transferred over to DNA, let me see if I don't think they show it on here. But once it's transferred over to DNA, this U converts over to a T, right? And that's through the transfer RNA molecules, right? So, and there's a bunch of different genetic codes that they have. Let me see. All right, whatever. I'm not gonna go into it. But just know you can go here, go to genetic codes, and it'll show you like all of the codes. And genetic codes are responsible basically for like what they call phenotypes or phenotypical things. And phenotypes are characteristics of DNA. So that's where you get like Caucasoid, Negroid, and Mongoloid from. And these are characteristics of different DNA types that are passed down based on the genetic code. But let's go back. All right, so boom. Now, if we go down to, we was at RNA right there, right? If we go down to transcription, right? This is going to tell you transcription is the process of copying a segment of DNA into RNA. The segments of DNA transcribed into RNA molecules that can encode proteins are said to produce messenger RNA, right? So this process called transcription is what creates mRNA, right? And this transcription is the process of it copying the segment of DNA into RNA, right? Let's see if it tells us what exactly does that. Copied into see all of these acronyms. ncRNA, right, which stands for non-coding RNAs, right? That refers to the genetic codes that are just passed up that they don't keep. One love to you, Ascendant Non-Ether and Henny God and Tees Lazaro and Or Heinzov, sending positive vibes to y'all for coming into the live. One love to you, Reflections. But like I said, there's a lot of these different things. And when I get into this, I'm going to start talking about this a little bit more right before we go. I'm going to show you how cloning is not something that you should be thinking is not going on. And it's not something that you should be thinking like is not a thing. Like cloning is clearly real. Like it's not like up for debate or nothing. Like cloning is done. I'm gonna show you in 2 seconds. Like not literally 2 seconds, but bear with me. Hold on. So let me see. So where was we at? And I could probably skip over some of this. I feel like you got the general sense of this, right? So now when you look at this, it's like, okay, I understand what this is. You know what I'm saying? These are guanine. This is thymine, adenosine, cytosine, and guanine. This is called a base pair, which is where they connect together at. Base pairs follow these base pair rules, right? And that means C go with G, T go with A. We know that thymine is directly connected to the gland that produces T cells and is responsible for your cells, protecting you, right? We know adenine is responsible for giving energy and allocating power to your cells to do things, which is why these two work together. We know that cytosine pretty much works hand in hand with guanine to make sure that everything is sent across, so yeah. Somebody says stem cells cured the third HIV patient today. I mean, listen, Dr. Sebi cured it with natural plants because it's not, that goes into viruses. You know what, since you brought that up, let's go into viruses right quick, right? Let's go into viruses. I was going to save that, but you know what? I feel like the energy is telling me to just do it. So viruses, right? Let's see. A virus is a submicroscopic infectious agent that replicates only inside the living cells of an organism. Viruses infect all life forms from animals and plants to microorganisms, including bacteria and archaea. This is us, by the way. Archaea is us, right? So let's go into this right now. Viruses. One of the things that I want to tell y'all about viruses, let's look at something called a provirus, matter of fact, real quick. And I'm gonna need to a provirus is a virus genome, right? And a virus genome just means a type of virus. That's basically what they mean. A genome is a set of genes that have characteristics. They're not going to let you, okay. A genome is all genetic information of an organism, right? So a virus genome, right? A virus's set of genetic information, right? So a provirus is a virus genome that is integrated into the DNA of a host cell. Now, the reason I wanted to bring this up, one love to everybody who came in the live, and I send a lot of love and respect for anybody who can stay into the live while we're talking about this type of information. Now, one thing that you want to keep in mind is that when it comes to HIV, right? HIV was a provirus, right? Now, here's the thing with proviruses that bothers me. Proviruses have instructions. Peace to you, Reflection. Proviruses have instructions. Let's see if they tell you, hold on, that is integrated into DNA of a host cell. In the case of bacterial viruses, proviruses are often referred to as prophages. However, it is important to note that proviruses are distinctly different from prophages, as these terms should not be used interchangeably. Unlike prophages, whatever that means, we'll look at it in a second. Proviruses do not excise themselves from the, yeah. So basically, they just saying like basically, the way a provirus works, a provirus is to the death. Because what a provirus does is it gets into your DNA of the host cell, and then it starts to create RNA. Right? And this shit makes me want to cry, breaks my heart to know that this stuff was happening to my people. They basically was infecting people with, look at what it says. Invades a cell. The RNA of the retrovirus is reverse transcribed into DNA. Right? What that means is this virus gets into your body, and then it tells your messenger RNA to tell your body to go kill itself. So you know how you say to somebody jokingly, hey, go kill yourself. This is what proviruses do, like HIV, right? Now, mind you, there's a lot of speculation behind that because HIV, wasn't really killing people like the treatment that they was giving people for HIV was. So, but anyway, that's neither here nor there. That would be a whole different lecture. But just based on what they tell you, then you want to look at this, right? Protein synthesis. We know that protein synthesis is basically the way your body articulates what you put in your mouth, right? So when you eat chicken or cow, the proteins, the enzymes act as a catalyst to tell you what to take from that meat. What they're saying is, let's see what it says. What they're saying is basically once you get infected with a provirus, it changes the machinery of this. It hijacks it to produce more viruses. So it basically tells you every time you eat something to turn that food into another virus. And this is where you seen those videos under the microscope where it had like a cell, and then that cell go to another cell, and then that cell split into another cell, and then those two cells go to other cells, and then they split into two cells. Because basically each cell is teaching through RNA the other cell to go kill the others. All right. It's like if you was to hijack somebody's child and teach the child to murder their parents and then send them back home. You know what I'm saying? They're gonna teach the other children to murder them, and then each child is gonna murder their parents. Now, it's not a good analogy because there's only two parents, and after one murder, it's the end. But you get conceptually what I'm talking about, right? All right, so let's go back. RNA pretty much speaks for itself. You know what I'm saying? We pretty much like know what it is, and then there's another process called translation, which is basically how it gives it back to the DNA. So in molecular biology and genetics, translation is the process in which ribosomes in the cytoplasm and endoplasmic reticulum synthesize Boy, they make me so mad with these words, but I be wanting to go slap. The cytoplasm, right? This is just the inner workings of a cell. You know what I'm saying? And this is like the external workings of a cell. They whatever, man. Anyway, just know that this is how it spits it back out and sends it back to the DNA. So, and now this one is a whole nother lecture. We getting close to the end of what I had planned, and then I'm gonna just show y'all some other stuff that I wanted to show you real quick. But within eukaryotic cells, DNA is organized into long structures called chromosomes, right? Now, this is where reflection be talking about how they don't have chromosomes like us, right? Because eukaryotic, well, let's see what they say. Before typical cell division, these chromosomes are duplicated in the process of DNA replication, right? Providing a complete set of chromosomes for each daughter cell. Eukaryotic organisms, animals, plants, fungi, and protists, store most of their DNA inside the cell nucleus as nuclear DNA, and some in the mitochondria as mitochondria DNA, right? Now, in contrast, prokaryotes, bacteria and archaea, right, store their DNA only in cytoplasm, in circular chromosomes, right? Within eukaryotic chromosomes, chromatin proteins such as histones, right? And just so you can see what these are, right? Chromatin is just the stuff that's in chromosomes, basically. Like, chromatin is a complex of DNA and proteins found. And proteins are what? Just macromolecules, right? Large molecules. That's all you need to know. Chromatin is a complex of DNA and protein found in eukaryotic cells, right? So it's basically the stuff that's in chromosomes. That's all you want to know about that. Now, histones are basic proteins abundant in lysine and arginine, right? Now, lysine is a protein that you can find in a lot of plants, but if you look up where do you find lysine, they going to see, this is where, listen, all right, y'all, listen. Follow me for a second, because I'm going to have to deviate, but I want to show you something. This is where, when reflection be talking, right? What does he say? They are eukaryotic cells, right? They are eukaryotic and we are prokaryotes, right? Now, within eukaryotic chromosomes, chromatin proteins such as histones, right? Now, one example, I did a lecture on protein when I explained the fuckery around proteins. But look at this chemical, lysine, that they talking about, right? Now, lysine let's go into it real quick. Let me see if I can pull it up. Where are you? Lysine, right? Now, lysine, right, is one of many amino acids, right? It's an amino acid. And you got to keep in mind that proteins are just groups of amino acids, right? And amino acids are proteins that basically send codons, right? Remember these codons? AAA? What's that? Adenine, adenine, adenine. And this is adenine, adenine, guanine. These are codes that mean things. But basically, what I wanted to show you was lysine, right? They told you that that was for eukaryotic cells, right? Now, if we type in, right? If we type in where can lysine, right, be found, what they gonna say? Meat, cheese, pork, all this shit that'll kill your ass, right? That's what they gonna tell you where to find lysine, right? Now, let's look at this, right? Let's look at something called pyro. And I showed this in another one. Pyrrolysine, right? Now, let's look at this. First of all, let's look at what pyrrolysine is. Pyrrolysine is located in the catalytic center of the proteins and is essential for their function. The energy generation process of the archaebacteria. You only find this in bacteria. That's what I'm trying to show you. Would not work without pyrrolysine, right? What they're saying is pyrrolysine is necessary for bacteria. Now, where do we find pyrrolysine? Pyrrolysine is synthesized by joining two molecules of lysine, right? So basically, one, this just says so much. This was created. Like, you just got to understand, like, how they know that. Like, you know what I'm saying? Like, this was created. And on top of that, this is what they're telling you. Let's go back to the drum board here. This is what they're telling you is what you need in your body. Let's go to it. Look, I'm gonna jump to it real quick. All right? Because I did another lecture on this, but I don't know if people are going to look at it. So in case somebody's actually watching this that can learn from this real quick, let me show you something. Let me show you something here. Hold on. All right, protein. What is protein, right? Amino acids, right? Proteins are just long chains of amino acids, okay? That's all you need to know. It's a long chain. So let's go to amino acids, right? What are amino acids, right? Now they tell you that there are more than 500 that are naturally occurring, right, in your body. However, 21 of them, where's the word 21? I read this already, I just don't remember. They say only 20 appear in the genetic code, right? Which means the other ones are what? Inorganic or probably not real, but or they can't find a way to create them. But look, this is what I wanted to show real quick. If I can get to this chart. Where are you, chart? Come on, don't play with me right now. I'm deviating from something else that I'm not even supposed to be deviating from. And like, you're going to make this hard for me right now. Where's the CHART? Hold up, y'all. Because I think it's like amino acids, blah, blah, blah. Where is it? Is it on protein? Hold on, y'all. Give me 2 seconds. All right, so look. I'm not going to go into it right now because I would have to find it. But just understand that proteins, these proteins that they're trying to tell you, these tie back to bacteria. And the areas that they tell you to get them in are found in bacteria created things like cheese. Cheese literally comes from bacteria from milk, pasteurized milk. You know what I'm saying? Let me see if you can find, hold on. So look, right, if you type in what vegetables can lysine be found in? Give me 2 seconds, y'all. It's hard for me to type with this phone in my hand. Believe it or not, I type 70 words per minute. But when I got this phone in my hand, they keep taking you back to this. So listen, this is what I'm saying. Why would you need this? None of this is natural. We never ate this. We don't eat meat. We don't eat red meat, pork, poultry, cheese, certain fish, eggs. We didn't eat that. So how are you telling me that this is necessary? This is a necessary protein. This is one of the ones they tell you that you need in your body. You know what I'm saying? This is how you understand where the lies are. You know, there's lies up in this joint. You gotta just like pick through it. But like I said, it says they are found in eukaryotic cell nuclei, right? We don't have them in ours because it actually says that you can't. If you look at this, it says that you can't naturally produce it. Let me see. Amino acid, biosynthesis, and protein contains like almost amino acid, kind of lysine referred to for the purpose of. Hold on, y'all. Okay, here we go. The human body cannot synthesize lysine. I repeat again. The human body cannot synthesize lysine. Now, why in the world would you need something that you can't synthesize? You see, and what this does is this creates a little bit of what we call a, see, and look what they say afterwards. It is essential in humans and it is essential. Oh, here's the chart, y'all. Here's the chart. I bet you this is where the chart is that I saw. Ha, what I tell you. See how the universe will bring you to it? So look, they say that these are all essential. Bro, we just read that your body can't even your body can't even synthesize this. Why the hell would you put this in your body? And look what's over here. We know what that is, right? Which is what? It says it's non-essential, but it don't tell you what it is, which is bacteria. Like when you look at this, what does it say? Pyrrolysine is an amino acid that is used in the biosynthesis of proteins. Synthesis, think about synthetics. Think of processes with living organisms. That's what bio means. Living organisms, synthesis, a process with living organisms. So an amino acid that is used in the biosynthesis of proteins, which are groups of amino acids, and some methanogenic archaea and bacteria. It is not present in humans, right? So this is not present in humans, but yet two of these compounds together make this, and this is essential for my body. Are we all feeling, we all getting what I'm saying with this? So this is essential to my body. We know what essential means. You die without it. Lysine is essential to my body. It's a protein that you need, that they tell you to get it from where? Meat, dairy, cheese. They tell you ingest all that crap because you need this. But first of all, this can't even be synthesized by your body. And on top of that, it creates this, which only exists in bacteria. So how do you not get sick? I'm trying to figure out how can you eat that stuff and not, look, you, hey, God in the treats, you're 1,000% right. You're 1,000% right. I just don't want to go there, but you're 1,000% right. Be the God sending positive vibes to you, Reflection. I appreciate you for coming in. All right. So let me go back. This is just to add another reason, you know, and look, these, if you was to go through these, look at these. This is what I was talking about when I said 21 earlier of the 20. Look at this. All right. An essential amino acid is an amino acid that cannot be synthesized from scratch by the organism fast enough to supply its demand and must therefore come from the diet. So that means you telling me that this stuff, my body can't make it fast enough. So the only way I could get it is to get it through my diet. Of the 21 amino acids common to all life forms, the 9 amino acids humans cannot synthesize are these, right? Including lysine. Now, if you look up all of these, and I'm going to say you the time because I did it already. If you look up all of these are going to tell you to eat meat, dairy, pork, eggs. That's all they're going to tell you to eat. And these are the essential that you need. I don't eat none of that shit. And I never will. Not anymore anyway, you know what I'm saying? So, yeah. And then they have some other ones they say are conditionally essential. Listen, this whole thing is just so that doctors can tell you to eat that shit. This is so that cause doctors don't know shit. Like half the time all doctors know is what was put in that goddamn curriculum for them to know. They ain't no smarter than no other doctor. And if they are, they gonna end up dead. Period. Because this is one dragon with 1,000 heads. This is what we gotta understand. One dragon with 1,000 heads, baby. That's it. Alright, let's go back though. I just wanted to touch on that real quick, y'all. Because I seen that pop up and wanted to touch on it. But you gotta understand that, like they said, eukaryotic organisms, right? They have the ability to get it from there. Like if you look at eukaryotes, let's look at eukaryotic organisms, right? Let me see real quick if they tell you. Cause they say in here that they have, first of all, look, the eukaryotes are usually now regarded as having emerged in the archaea, or as a sister of the now cultivated Asgard archaea, right? And when they talking about Asgard, they're talking about Thor, Loki, Heimdall, you know what I mean? They're talking about those, which are basically types. And I'll show y'all that real quick. It's not going to be under eukaryote because it doesn't fall under them. But if you type in prokaryote, right? Just to show y'all real quick. Some of y'all seen this already. Right? You type in prokaryote, you're going to see something here, right? Where they tell you there is increasing evidence that the roots of the eukaryotes are to be found in the Archaean Asgard group, right? And then when you go to Asgard group, right? For those of us who are fans of Marvel, or somebody corrected me and said Greek lore, whatever you want to call it. I'm using Marvel because that's more notable. Like come on now. All of it is based on that, you know what I'm saying? Greek mythology. But anyway, Asgard, right? They call it Asgard Archaeota, right? Is a proposed superphylum. For those who don't know, a superphylum is a classification. It's just basically saying like how something is classed, right? And they also call that taxonomic rank, which is how they rank you, right? This is the ranks: life, domain, kingdom, phylum. They saying it's called a superphylum in between these, species, right? So let's go back. But they says consisting of a group of archaea that includes Loki Archaeota, Thor Archaeota, Odin Archaeota, and Heimdall Archaeota. Now they don't go into a whole bunch of detail about this when you go to it, because it seems like they just really don't know. But like they just tell you something about, let's see what it says. Loki Archaeota is a proposed. See, proposed. Anytime they say proposed, they don't know what the hell they talking about. Because they wouldn't use a word like proposed if they were sure. Trust me. Loki Archaeota is a proposed phylum of the Archaea. The phylum includes all members of the group previously named Deep Sea Archaeo Group. And see, DSAG is just an abbreviation that they created for a marine group that was studying, right? So basically they found these underwater, they found them in all kind of like conditions, right? And these things can survive and all. See, they said it was taken near a hydrothermal vent, right? So basically these are, the reason that I'm showing y'all this, that I'm talking about this, is because all life forms are divided into 3 parts, right? You can see they tell you right here, right? All life forms are divided into 3 parts. Hold on, let me go back to the last one because they tell you on eukaryote a little bit more clearly, right? The domain eukaryota makes up one of the 3 domains of life, right? And when you look up the 3 domains of life, that's this phylological tree, right? The phylological tree will show you all domains of life. And they are broken into 3 domains: archaea, bacteria, and eukaryote. Now, archaea is the same thing as prokaryote. They just started calling it prokaryote instead of archaea. And you can see that when you click on prokaryote, and it has like archaea in parentheses and stuff. Like, I'm not going to go into it. You can see it. All right. But the reason I'm saying that is these are the 3 domains of life. That means everything that's alive is one of these things. It's bacteria, which is normal. We know bacteria is normal. There's nothing wrong with bacteria. You know what I mean? Hand sanitizer was a absolute assault on bacteria. We have bacteria in us, and these are living organisms that help us with biodegrading. They help us with, certain cycles like the carbon cycle and things like that. So there's nothing really wrong with bacteria. There are some. See, look, bacteria are ubiquitous, meaning every damn where, mostly free-living organisms often consisting of one biological cell. They constitute a large domain of prokaryotic microorganisms. So these, with their basic, peace to you, Reflection. Shout out to everybody in the comments. One love to you. I appreciate you for coming in. So basically, just know bacteria ain't that bad. The only reason that there is bad bacteria is because you've had evolution take place of bacteria because of their atrocities, right? So if you have regular bacteria that breaks down an apple and degrades it so that it can be, able to be digested by the planet in the soil, and then you take and clone an apple and make a fake apple that has other things in it, now you're going to have different bacteria. Now you're going to have bacteria that grows and wants to attack different things. And you're going to have, you know what I'm saying? This is where germs come from. This is where all of this comes from. It comes from the unwanted and unvalidated, nosiness of those who are not indigenous. It comes from the meddling of those who are not indigenous in the affairs of those who are indigenous. And this is how you get mutations. This is how you get all kind of crazy things that are due to, different changes in the environment and everything. But I can do a whole lecture on eukaryotes and prokaryotes and bacteria, the 3 domains of life. I think I will, because it would actually help me too. But they don't know a lot. To be honest with you, when you start talking about prokaryotes, they don't really know a lot. Like, they'll tell you that they do know some, but they'll tell you like there's things that they don't know about. There's energy forces that they're not sure of, how this is happening, and all these kind of things. And you'll see that prokaryotes are very similar to plants. Like, they're basically plants. Plants are prokaryotes, and we are prokaryotes. You know what I'm saying? And then you see they got some other things for eukaryotes. Let me go back, y'all. I got all sidetracked. Hold up. So look, right? Just keep in mind that chromosomes, there's 2 types of chromosomes. So cause we do, you always hear them say, you'll hear people say Caucasoids don't have chromosomes. We all have chromosomes. What happens is the structure of chromosomes is different. So we have what we call a circular chromosome. See, they have something called a linear chromosome, and they have something called mitochondrial DNA, which is like stored within a cell, whereas ours is stored in chromosomes, right? So a circular chromosome, which occurs in bacteria and archaea, right? And mitochondria and chloroplasts. These are basically plants, right? It in the form of a molecule of DNA. Now, the reason that it's circular, they'll tell you this. See how it says unlike the linear chromosome of eukaryotes. Now, they'll tell you that the purpose of this is because of the amount of, they'll tell you this basically because of the amount of information that has to be sent around. The amount of information that has to be sent around, it has to basically like come back around to itself. It's hard for me. This is why I'm saying I have to do a separate lecture on it, because it's also some things that I need to, to understand about DNA replication, how it relates to linear chromosomes. Cause I don't understand how it comes back if it's linear. Like that's something that I'm not going to waste y'all time with. A chromosome which in linear shape and contains a terminal end. Yeah, see, their chromosomes have terminal ends. This gets into genetic coding. Coding at the end of a genetic code. Remember when we was looking at it earlier, y'all? In Nasia, give me 1 second. Remember when we was looking at it earlier and we saw like UGA, UUU, UCT, AAG, the coding? There's something that comes at the end of that called a terminator. And this is actually a metaphorical explanation of what the movie Terminator was about him getting rid of John Connor so that he didn't keep coming back and replicating. Terminator is what they refer to as the closing of a DNA code or a genome code, as they call it. Let's go back though. Right, we don't age, but there's things that are put in place that make our bodies react. Like ozone, which is an oxidation chemical meant to oxidize you, and oxidation in itself causes aging. So, oxidation is a electron thief. You know what I'm saying? And, Nasia, when you said, is this an encyclopedia? Yeah, this is Wikipedia, right? So Wikipedia, also known as the free encyclopedia. I use Wikipedia because honestly and truthfully, there is no better option than Wikipedia. I mean, listen, I've heard a lot of people say, I use this. Well, I use book. You can also use Britannica. Britannica works good too. I use them sometimes. Let me just show y'all real quick so you can see it. Oh, my bad. This is an order we was doing for somebody with the chakra thing. What's it called? It might be called Britannia or something. Hold on. I'm mad I'm wasting time with this, but I just want to address what you're saying because it is a valid question and it's something that a lot of people, yeah, so Britannica, right? Britannica Learn is a school that you can go to, or it's an encyclopedia, Britannica.com. And this works just like Wikipedia, except it's not as detailed. See, the thing about Wikipedia is that you can go into Wikipedia to learn one thing. Look at this. Y'all seen everything. We've been talking for hours now, to the point where my phone just popped up and said I got 15% battery life. We've been talking all of this time, and this all came from one page. Just this right here. Just from here up. Because every single link is a whole nother lecture. Because it take you into everything else. And see, Wikipedia works like, the reason that I'm gonna get off this in a second, y'all. My bad. We're gonna get back to the lecture, but I just want to explain something about Wikipedia because Wikipedia gets a lot of bad rap, and so does everything else that will actually teach you and help you out. Of course, they're not gonna send you to something that's gonna help you. Any kind of school, any kind of scholastic center, anywhere that has a curriculum, right? All of these are code words for autocratic societies or dictators. Any of those, they're definitely never gonna send you here. Because you're going to actually learn if you go here. And they don't want that. And you're going to learn at a rate that's going to be beyond anything else you could. Because listen, you can buy a DNA book, right? You can buy a DNA book. But a DNA book might not necessarily break down to you what covalent bonds are. You're going to have to go get another book for that, right? And it might not break down to you a sugar phosphate backbone. Or base, no, it might break down base pairing rules, but it's not going to break down to you nitrogen cycles, right? You could go into this, and this is, look, the sequence, it's not going to, listen, look at this. Just from this one link right here, it is the sequence, right, and they talking about the sequence of the nuclear bases. I click on that, it bring me to this. So now I'm learning about the nucleic acid sequence, right? Guanine, acid, adenine, cytosine and uracil. You know what I'm saying? What I'm saying is when you jump on here, you learn so much. This is 100 books right here. This is 100 books right here. Now, people are also going to say, well, the problem is you don't know where it came from, right? You don't know where it came from, right? And this is why I said earlier when I was showing people, you got to pay attention. So like, let's say I read this, right? And I said, chloroplast DNA, and somebody like, ain't no such thing as chloroplast DNA, what you talking about, blah, blah, blah. This is where I would hit this little number right here. You see where's my pointer? See there's one right here next to uracil, right? And that comes from a book that you can read, right? You can click on the link. And then there's one right here next to chloroplast. This is a book written by some dude named Russell P. or he's a genetics, whatever. You can click this and that will make you feel better. You know what I'm saying? Cause some people, they like, man, I don't feel right reading this here. Okay, well, go read it where Russell wrote it the other place. You think the same people who own this ain't the people who own the publishing companies that publish his book? You think they're gonna let him, like, come on now, you just gotta put two and two together here. You just make stuff hard for yourself when you wanna go find 20,000 books and have a whole bookcase of all of this stuff when I can click a link and find out more about it than you even know. Literally, like, you know what I'm saying? And they got a section on here, right? Goddess, I want to make sure you know about this. They got a section called references, further reading, and external links, right? References, this shit so much I don't even like to look at it because it make my hair hurt. But look at this. These are all of the books that all of these knuckleheads gonna tell you not to look at Wikipedia because of, right? Here go the Russell one that we looked at, genetics, right? These are books. These are codes that are assigned to books when you sell stuff online, if y'all familiar with that. So that's why it looks like that. But you can go to any of these. Look at this. Deoxyribose. They even tell you where they got the definition. You know what I'm saying? So anybody that tell you, oh man, I don't believe. Look, they even got PDF files. They even got PDF files from actual doctors from 1953 showing you this stuff. Ain't no book gonna give you that, bruh. Come on now. Don't talk to me about a book. Like, if you talking to me about books, like, honestly, that's like you paying your rent and mail. That's like you paying your cable bill while sending in a check. Like, it's 2000. Like, I'm not doing that, bro. All of the books, even if I was to get some book because it made me feel good, I would just pull a book up on here. You know what I'm saying? Like, I'm not gonna, so that's also an excuse that a lot of people use for them not to look up information and learn stuff, because they like, oh, well, I gotta get these books. I don't got the money for the book. Because you know how bad I would like a biochemistry book, a DNA book. You know what I'm saying? I would love a DNA book. A book that say this on the front of it, and a book that say this, and a book that say all of this inside the book, so that I can hold it inside of Starbucks like this. You know what I'm saying? And make everybody think I'm smart because I'm reading it, right? That'd be cute, right? That's cute. You got a book. That's cute. But what I'm concerned about is information. I really don't care about your book and your hardback. Like, at the end of the day, I care about information. And those references that I just showed you want to check. There's also further reading, right? So that you can look up more. Right? So these are external links that take you to different sites that teach you about electron microscopes, DNA learning centers, double helix, right? This will teach you about them double helix structures that I was telling you about, the A to B to Z, right? All of this stuff can be found. ENCODE, right? This stands for, ENCODE stands for, of, what is it? Encoding of, it stands for like the encoding of, what does this stand for, yo? Cause I looked this up. This is like a group that they have where they keep up with all of the codes. Alright, my bad, y'all. I ain't gonna go there. But this is a website. This is another website, nature.com, that you can go to. You know, for the people who like, well, why Wikipedia? I don't like that, alright? So that was just my response to you, Reflection, Goddess, who said that, because I feel like a lot of people don't learn stuff because they shy away from Wikipedia because they was told by the ops not to look at it. You know what I'm saying? Nobody's going to tell you to look at something that they know is going to give you information, especially when they trying to like teach you otherwise. You know what I'm saying? So look, so that's pretty much, hold on, let me plug my phone up before it die. Number one. Oh, I appreciate you, Nasia. I appreciate you, you know what I'm saying, coming in here and being in here with me. Could have been anywhere in the world, but you in here with me. The internet in general, as much as you can now, because we'll be cut off from these sources. Exactly. Yeah, exactly. And all of this information is right in front of your face. I was just scanning the comments for everybody who in here. One love. Shout out to Big Heart. One love to you, Reflection. I appreciate you for being in here. Big Heart always got so much heart, man. Listen, his name speak for itself. You hear me? So, and you can watch it back, Big Heart, if you want to. We just went over some of the things that, and it's funny, Big Heart, that you come in because, you know, you watch Kalai. A lot of us that are in here right now, we watch Kalai. And this is all of the stuff that Kalai talks about. This is all of the stuff that he talks about. But it makes more sense to you when you know what these things mean. You know what I'm saying? He was just talking about adenosine, right? If you remember, he was talking about adenosine. What's that? That's adenine. This is one of the nucleobases of your DNA, and it contains nitrogen. If you wonder why the hell nitrogen is in there, it's because nitrogen has, it works alongside phosphates, right? This is why when they tell you a nucleotide, they tell you a nucleotide is basically a nucleoside without phosphate, right? See? A nucleotide is a nucleoside and a phosphate, right? But phosphate is orthophosphate is a derivative of acid. It's dysfunctional group, right? So, you know, at the end of the day, they have to get nitrogen to work with that, because when you look at nitrogen, like I said, nitrogen, I'm not going to go into it, but nitrogen has something called the nitrogen cycle, right? Where I keep on saying I'm not going to go into stuff. What kind of lecture do you say I'm not going to go into stuff about? Hold on, let's see if they show you down here. I think it's down here somewhere. See, when I be learning a lot of this stuff, to be honest with you, it'd be 2 a.m. and I be in my bed, I be on my phone, so it look different. You know what I'm saying? Like on the phone, it look different. That's why sometimes I be like, wait, where is it at on here? They got something called the nitrogen cycle, right? Organic, hold on real quick. Nitrogen is one of the most important elements in organic chemistry. Many organic functional groups involve carbon-nitrogen bonds, such as amides. This is where your amino acids come from. This is in amines, right? This is where aminos come from, amino acids. But hold on. They got different names. Fuck they words. They basically change the word every time they restructure the way the compounds are. But what I'm trying to get you to see, hold on, that's not even what I'm talking about. There's something called a nitrogen cycle, bro. Why y'all playing with me right now? Like, I'm gonna have to start, see, because when I go over this stuff, I don't like, you know what I'm saying? I don't like record it or write nothing down or nothing. I just be like, remembering what I learned. Alright, well, I don't see it here, but there's something called a nitrogen cycle. And it's also a carbon cycle as well. And those two are related. And this is why you need nitrogen. But let's go back. I ain't even going. And look at this. Hold on, let's go forward real quick. Look at this shit. The name nitrogen was suggested by French chemist John. You see why I say fuck they names? You see why I say fuck they names? Because the name nitrogen was suggested by French. And what French mean? French mean I killed all your people and took your land from you. That's what French mean. Suggested by French chemist Jean-Antoine Claude de Crafetal in 1790, when it was found that nitrogen was present in nitric acid and nitrates, right? Now, when you look into, now maybe we'll get into it. Hold on. Because this might take me to where I seen it at. I just want to show y'all the nitrogen cycle real quick. That's literally all I want to. Is that pee in a bottle, nigga? What are you? Goodness gracious, yeah. Okay, all right, forget it. I'm giving up on the nitrogen cycle. But the point of the matter is, I wanted to mention to you, suggested instead of azote. Bruh, why'd you turn on my frequency music? Like, what? My bad, I was playing frequency music and it just popped on. Maybe they want me to turn it on. I'm gonna turn it on in the background, y'all. The universe made me click that. Like, I don't know why it just popped up. So, I'm gonna play the love frequency in the background. If it presents an issue, if you hearing me, let me know, but I'm usually loud, so. But look, they said this guy, he suggested the name instead of the name azote, which is what nitrogen was originally called from ancient Greek, right? So what this means is, during the ancient Greek times of ancient Greece, they already knew about this. You see how like how this shit like all come together, bro? They knew about this since ancient Greece. And this is where us teaching them comes from. Look at this. Oh, bro, look at this. Hold on. Let's get into it real quick. Hold up real quick. Cause peep this. Nitrogen is a non-metal and the lightest member of group 15 on the periodic table, often called the nitrogens, right? It is a common element in the universe, estimated at 7th in total abundance in the Milky Way and the solar system. Look at that. And y'all know what the most abundant thing is? Or like the second most? Hydrogen. Right? Which kills me because hydrogen heals you, and they actually tell you. But what I'm saying about this is, just look at certain terms. Ancient Greece, Milky Way, solar system. They know more than they telling you, bro. Like, this is what I'm saying. Like, they know so much more than they are telling us. But we don't need them to tell us because we already know. Let's move on. Somebody said, and Greece got their knowledge from us. Exactly. Yeah, exactly. Alright, so let's go back. I don't know where I was, y'all. I think I pretty much was at the end, just basically saying at the end of this. Now, there's much more under this, right, that I wanted to show y'all. And I'm trying to remember what certain ones was, right? So, like, when we get into, you know, hold on, bear with me for a second. Bear with me. So, I know I wanted to mention this to y'all, right? DNA molecules, actually two polymer strands bound together, covalent bonds, double stranded. Okay, hold on. Let's do. So look, right? Remember how we was talking about your GC content? And GC are the base pairs what? Class. It's like, yeah, I'm just messing with you. Guanine and cytosine, right? So the stability of the dsDNA, right, which is double strand DNA, right, depends on not only the GC content, but also the sequence, since stacking is sequence. Okay, and also length, right? Now, I say all this to say, or I just said that to say, and this is what it looks like when they pair them, right? These are the base pairs where the hydrogen bond is, right? Boom, boom. That's the hydrogen bond. Boom, boom, right? That's the lines that you see connecting when you see the whole spiral thing. The more of this you have in you, the stronger your bond is going to be. So those 4 nucleobases are very important, because you can take that alone and run with it. Eat a bunch of shit or find some way to generate that chemical inside you and raise it. But anyway, hold on, y'all. Give me 1 second, because I wanted to show y'all something else. But I did want to take a second to say one love to everybody who's in here and everybody who stayed in here and who managed to watch the live. Even if you caught the end of it, go back and watch the beginning if you like, to get anything that we might have touched on in there. You know, but yeah, let's get back. Alright, so like, so first of all, for y'all that don't know, the reason that you need, the reason that you need to eat alkaline is because alkaline is the opposite of acidic, right? Which means it can bond well to hydrogen. That's really what you want to know. It can bond well to hydrogen. And hydrogen is what we are made out of, right? It's not what we're made out of, but hydrogen is like, it's the most ubiquitous substance that is a gas. The sun is made out of hydrogen or surrounded by hydrogen. You know what I'm saying? Hydrogen makes up like 80% of the universe, they were saying. So like at the end of the day, it's very important. And pH stands for potential hydrogen, for those who don't know. So when something has a high pH level, that means it has a high potential to bond and connect to hydrogen. And you want to understand that hydrogen is what? Hydrogen is what bonds your DNA. Right? So when you eat things, one love, reflections, one love. When you eat something that has a high pH, the chances of hydrogen bonding goes way up. But when you eat things that are acidic, it goes down. So the chances of you having what they call clusters or what they call errors in your genome codes, right, that are being sent through are higher if you eat acidic things. That's why you're more likely to pass down a disease. So like if you, so y'all know this DNA structure exists in sperm cells as well, right? That's part of the RNA's job, to make sure that it exists in sperm cells. So if you got highly acidic sperm cells or highly acidic DNA cells in sperm, there's going to be some problems when it's trying to code, because the bonding, the pH level is too low. You understand what I'm saying? This is why, like, it makes more sense when you explain something scientifically than just saying, yo, you gotta eat alkaline because the pH level high. Like, no, bro. pH stands for potential hydrogen, and hydrogen is literally what connects your DNA. Hydrogen bonds. That's why you need a phosphate to mix in with the nucleobases, right? So anywho, let's go into something, right? I wanted to show y'all. I think it was under damage. Was it under damage? Hold on, y'all. Give me 2 seconds. Oh man, I'm gonna show y'all something about this shit too, but I wanted to show y'all how cloning is real. Like, and there's no, I mean, we all know cloning is real anyway, but you know. So look, let's get into that. Matter of fact, uses in technology, right? This is telling you how they use DNA in technology. Why the hell are you using DNA in technology? But whatever, right? Methods have been developed to purify DNA from organisms such as phenylchloroform extraction. What? My bad, y'all. Extraction, and to manipulate it in the laboratory, such as restriction digest, right? And the polymerase chain reaction, right? Now, when they say restriction digest, think of restriction, which means to hold shit back and restrict it, and think of digest, which means to process. It's what enzymes do. It's what catalysts help your body do when you eat things, right? But let's see what they say. A restriction digest is a procedure used in molecular biology to... My bad, y'all. I got this gnat. It's annoying the hell out of me. And molecular biology to prepare DNA for analysis or other processing. It is sometimes termed DNA fragmentation, right? So basically what that means is they are splicing it. And that's an actual term, splicing DNA. They talk about it. But let's go further. Polymerase chains, right? Let's look at polymerase. Hold on. Let's look at polymerase. Where's it at? My bad. Hold on, y'all. Gee, my computer is... Why are you pulling up a bunch of crap? Alright, I don't know what's going on here. Polymerase, right? And we know what poly means, right? It means many, mers, many parts, right? A race, right? Let's see what they're saying, though. Polymerase is a method widely used to rapidly make millions to billions of copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail. Now, so far, we haven't even gotten through this shit. So far, we know that they found out a way to splice DNA, meaning take sections or fragments of it, and they found out a way to study it and do shit with it, right? Cause chemical reactions with it, right? So that's the two elements that you need right there. But let's go. Yeah, we getting there. They use DNA and AI. We getting there. That's where I'm going with this. But I guess y'all already woke a little bit, so I can like get there with y'all. But basically, modern biology and biochemistry make intensive use of these techniques in recombinant DNA technology. Recombinant DNA. Now look, they tell you, this is a man-made DNA. Look at that. Man-made DNA sequence that has been assembled from other DNA sequences. They can be transformed into organisms in the form of plasmids, right? Let's see what plasmids are, right? Plasmids is a small extrachromosomal DNA molecule within a cell that is physically separated from the chromosomal DNA and replicate independently. Now let me ask y'all something. For those that have been paying attention and following me, right? Right? And been following me, right? What do this sound like? I'm going to answer what I think it is. A provirus. A provirus. A provirus, right? Proviruses have their own sets of independent DNA. And it comes with instruction. You got to understand what DNA is. So it's instructions for your body to do shit, right? So basically, here they're telling you that they can take fragments of DNA, to cause chemical reactions and make millions and billions of copies of it, right? And then recombine them. They just told you. They just told you right there that they make clones. And then they said man-made DNA, right? Now here's what I'm saying, right? They can be transformed into organisms in the form of plasmids or in the appropriate format by using a viral vector. A viral vector. Motherfuckers. Viral. Yep, COINTELPRO. That's exactly what it is. Infiltration. Viral vectors are tools commonly used by molecular biologists to deliver genetic materials into cells. Oh my God, bro. This process can be performed inside a living organism or in cell culture viruses. Or in cell culture viruses have evolved specialized molecular mechanisms to efficiently transport their genomes inside the cells they infect. Can I ask y'all something? Isn't this exactly what we just read that a provirus was? Am I tripping? Am I tripping? Isn't this what we just read that a provirus was? How we figure out how HIV was made on Wikipedia? So, this is the shit I'm talking about, G. This is the shit I'm talking about. Let's move on. Let's move on, right? You see what they even say? The genetically modified organisms produced can be used to... What the hell is... Yo, why is my computer... Listen. I don't know what's going on with my computer, but it's been acting weird. Like, I don't know what's up. Alright, let me see. Maybe I gotta move or something. I don't know. Alright. The genetically modified organisms produced can be used to produce products such as recombinant proteins, right? Proteins. There go that word again, right? And that's that word that they tell you need, right? You gotta have proteins. Everybody needs proteins. Oh, protein. Oh, protein. I gotta have the protein, right? And protein is what? Amino acids, right? Proteins is amino acids. A chain of amino acids. And what do amino acids do? Amino acids instruct your body on how to take information, basically. Somebody said they just got fired from their job. Oh man, hey listen, y'all do me a favor please, because I gotta go to the bathroom like super bad, but I really don't want to end this live. Like, give me 1 second, all right? Don't leave for those who... I mean, if you want to, leave, but if you don't want to, like, don't. one second, reflection. Love you. Alright, reflections. My bad. It was 18 people when I left. It's 17 here, so y'all love me. At least 17 of y'all do. One of y'all ain't like me. Nah, but listen. So, I just had to go to the bathroom so bad. I was holding it, and I was trying to hold off to the end, but I don't want to rush this because I enjoy this. Like, and you know, I enjoy talking to y'all. Let me take a quick look at that. There's some more I want to show y'all real quick, but I just want to take a look at the comments out of respect for the people who love me enough to stay in here. I'm definitely going to save this as long as they let me. Bastards, they better let me. No limit, you got this, you will get a better one. Yeah. No limit. Take it as a sign from the universe. That job probably was. Yeah. Nobody worry about no job, man. Don't Yeah, don't let that bother you. You know, I don't know what your personal situation is, but you know what I'm saying? I send you positive vibes, you know, and I just want to say to you, like, you know, try to work on whatever it is that you might be good at, you know, to make a business out of, you know, and just try to manifest, you know, try to work on manifesting. I know it's tough and that's easier said than done, but like, this is coming from somebody with 4 kids and no family to help them that take care of myself. You know what I'm saying? Like, so, you know, if you don't have kids, you already listen, people who don't have kids, I don't even know what you're complaining about. Like, because if it was just me, bro, I'd grab a book bag and be out there. Like, what you mean? I'd be a straight nomad. But I got 4 little ones that I'm taking care of, and that's in, you know, 2 of them in virtual school. The other 2 I'm fighting for, and you know, I'm fighting to raise correctly and everything. But look, let me get back to this, y'all. Let me get back to this. And what's good, Smooth 9 Ether Being? I appreciate you for coming in, Reflection. That was like a quick little, we took like a quick little interlude, if you will. I had to run to the bathroom real quick. All right. But yeah, like we was just saying in Reflection, just to summarize, we went over the fact that they use DNA and technology for genetic engineering, which is genetic modification, right? And they told us that they basically make man-made DNA through a process called restriction digest and polymerase chain reaction, which is where they basically create a chain reaction with the DNA that they took. So that's what they basically told us. They're not going to say it in those exact terms. You know what I'm saying? But when you see stuff, traveling has been a lingering thought in my mind. Yeah, I would be, listen, if I didn't have kids, if I didn't have my star seeds, there's no way I would be like in one place. Like, I wouldn't even be in a system. You wouldn't even know me. Like, literally, you wouldn't even know. You probably, maybe I would, but I wouldn't be like somewhere you could just find. I would be out and about, you know what I'm saying? Because I just don't like the, yeah, I don't like it. Let's go to damage. Oh, there's a section. Hold on, y'all. There's a section called damage, right? I'm trying to find it. Where is it? Because I remember there was something in the section called damage that I wanted to... All right, let's see damage, right? I think there was something I read under here that I wanted to... Oh, here's what I wanted to show y'all. Look, DNA can be damaged by many sorts of mutagens, right? And mutagens just refers to different mutations that happen during genetic code transfer, right? So, like, it can be... Yeah, it can be cancer, you know, it can be, I mean, but cancer is caused by them. Listen, there's no such thing as cancer. Cancer is caused by them. So when they say it's caused by the things that they give you to put in your body. So basically mutagens that are natural would be things like, you know, things that are specific to physiological changes in your body that result in some kind of a change that affects the genetic material. Anyway, my bad, y'all. Look, DNA can be damaged by many sorts of mutagens which change the DNA sequence, right? And the DNA sequence is what? The codes we was talking about, the DNA codes, right? It's the order of the nucleotides in DNA. It includes any method or technology that is used to determine the order of the 4 bases. And we already know that that's called what? Base pairing, right? It's called base pairing rules, like we went over earlier, right? So basically they just telling you that the base pairing rules can be messed up and you can have A with G, T with C, and a bunch of crappy stuff happen, right? But look what they tell you, right? Because they said mutagens was what? They said it was cancer, right? That's all they said was cancer there. But then let's go a little bit further. Mutagens include oxidizing agents. Oxidizing agents, right? Now, what is an oxidizing agent that we know of that we breathe in right now? Ozone, right? Oxygen, right? Or more so ozone. And ozone oxidizes. So when you go outside and you breathe in the air, it causes oxidation. Ozone itself is an oxidizing agent, which will mess up your DNA sequence. You see? It's right there. That's just one though. Then they say alkylating agents. I'm not going to go into that because alkyl, it's not even something that's real. Like, it's like, hold on, let me see if I can make it make sense why it's not. But look, Alkylation is the transfer of an alkyl group from one molecule to another. The alkyl group may be transferred as an alkyl carbocation, a free radical, or a carbanion, or a carbene, right? And this is some shit that gotta happen. That's why I said, like, this is inorganic, like, cause when they say carbanion, right? So there's these things called anions, and cations, right? Anion just means that there was a chemical reaction between two atoms and the amount of electrons went up, right? Anytime you have a lot of electrons, you have lower charge. A lot of people think electrons give you charge, like, because of the word electric, but electrons actually reduce charge. So the more electrons you have, the lesser the charge. And when you do a chemical reaction that results in less electrons, or more electrons, my apologies, that's called an anion. When they say carb anion, they talking about you using carbs. They probably talking about carbon, like when you doing carbon, you know what I'm saying? And then you got something called a cation, which is where the amount of electrons go down and the charge go up. That's how you get ionized particles. Or that, ionized particles just refers to, particles that have an electrical charge. And you need the amount of electrons to go down for something to have a charge. So anyway, let's keep going. So they're telling you that mutagens include oxidizing agents, alkylating agents, and also high energy electromagnetic radiation such as ultraviolet. We know that ain't about us, but X-rays. See, this is, now what do they always give us? They always giving us X-rays, right? They always give you X-rays, right? Penetrating form of high energy electromagnetic radiation. Most X-rays have a wavelength of 10 picometers to 10 nanometers. And 10 picometers is like what they measure, that's quantum physics. When you start getting into quantum physics and you measuring hertz and you measuring like how sound travels and stuff like that, that's what that's about, when you get into picometers. But that's neither here nor there. Point of the matter is, just be aware that when you get in X-rays, it's damaging you. It causes mutations in your genes. It mutates your genes. So imagine what happens when they say, oh, you got something wrong with your head, we got to do an X-ray on your brain. And they're sending these X-rays through your brain, known as X-radiation. You know what I'm saying? Now, how many doctors, if you have a headache or a stomach ache and you go to the doctor, is going to ask you if you had an X-ray recently? You know what I'm saying? How many of them going to ask you that? Not none. Not none, because doctors don't know shit except for what it's like religion. Doctors only know what they told to know in that damn book. It was something else. This DNA polymerase, man. DNA polymerase is a member of a family of enzymes that catalyze the synthesis of DNA molecules and nucleoside triphosphates, the molecular precursors of DNA. Alright, so you know why this is? Remember I was telling y'all about cDNA earlier? When I said cDNA, this is the one I was talking about, complementary DNA. cDNA, right? Not to be confused with cDNA, which is a different DNA type. But cDNA is synthesized from DNA and is created as complementary as a backbone, right? Look at this shit. Y'all didn't even see that. DNA synthesized from a single-stranded RNA template in a reaction catalyzed by the enzyme reverse transcriptase. cDNA is often used to clone eukaryotic genes in prokaryotes. Did y'all see that? It's like you'll be just, listen, it's like if you was reading a book and somebody was like, Yeah, so you reading a book about a murder or something, and you reading a book, and they like, yeah, so I went to the store, and then I blah blah blah, and I came back in the house, I grabbed my gun, shot her in the head, and then I walked in the other room and got you like, what you go back? What you say? cDNA is often used to clone eukaryotic genes in prokaryotes. Now, what they say earlier? What did they say earlier when we looked up prokaryotes, right? Hold on. Yo, because they be tripping me out. Hold on, let me show. Look. What did they say earlier about eukaryotic genes, right? Eukaryotes are usually now regarded as having emerged in the archaea. So, in the archaea is prokaryotes. Those two are interchangeable. So listen to this. The eukaryotes are usually now regarded as having emerged in the archaea. Right? So that's where they saying they come from. But then, my G, we turn around and 2 seconds later, you tell us that in a reaction catalyzed by the enzyme reverse transcriptase, cDNA is often used to clone eukaryotic genes in prokaryotes. Yeah, make sure, definitely look it up. So look it up, wow, so that we all can look into this stuff, because this is just like a brief glance over this. This is so much deeper than I'm even going. Each one of these I don't even go into them to show you. But look, a reverse transcriptase, RT, is an enzyme used to generate complementary DNA from an RNA template, a process termed reverse transcription. Reverse transcriptase are used by viruses. The more you learn, the more you see, boy, I tell you. Who you think made it now? Who you think made it now? So that ain't even what the hell I wanted to talk about. I was trying to show y'all this abomination, and I happened to see that and clicked on it. See what I'm saying about Wikipedia? You think all this would happen if I was reading a book that would be talking to me about DNA? You know what I'm saying? It would have been talking to me. We'd have still been reading about RNA and all the intricacies of RNA. And we don't really need to know that unless we're trying to fuck shit up. Which is why they want to know so much about it, because they're trying to fuck shit up. And they're trying to do inorganic, things with it. Not trying. They already have. They already do. But look, let's look into this. This abomination right here, right? So look, a DNA polymerase is a member of a family of enzymes, right? And enzymes are just catalysts. They just like protein, enzymes are protein. You see these words, man, they are interchangeable. That's why I say fuck they words, man. They just make these words up, whatever. Enzymes are proteins that act as a biological catalyst, biocatalyst. Catalysts accelerate chemical reactions. They basically like instigators. You know what I'm saying? When it's about to be a fight in a room, and you like, yo, he said your mother ugly. He said your mother stink. That's what enzymes are. That catalyze the synthesis of DNA molecules, right? Now, when we look up DNA polymerase, right? Hold on. I want to look up. Hold on. Give me 2 seconds. You see, and now this is just to show y'all like a reason why. So see what they say, before replication can take place, right? When they replicate in DNA, an enzyme called helicase, right? Which we know is what? Just a protein. It's an enzyme. It's a bunch of amino acids, which have the purpose of what? Just telling your cells what to do. They're instructors. Helicase, but let's see what they say it is, because I ain't never look at this. My bad. Helicase are a class of enzymes thought to be vital to all organisms. Thought to be. They don't say thought to be about stuff they know. This is bullshit. Their main function is to unpack an organism's genetic material. Helicases are motor proteins that move directionally along a nucleic acid phosphodiester backbone, right? And phosphodiester, they're talking about phosphate when you a derivative of phosphate. It's like an ester, which is a group of compounds that come out after you do something. Just this is what I'm saying. Like, if you look into it, you'll see more. But basically, what I wanted to show you here was before that process can take place, this unwinds DNA molecules from its tightly woven form, in the process breaking the hydrogen bonds between the nucleotide bases. This opens up or unzips the double-stranded DNA to give two single strands of DNA that can be used as templates for replication in the above reaction. Now, peace reflection to everybody in here. Leaf Love 9, I ain't seen you in a while. I appreciate you for coming into the live. Reflection, one love. Now, when we take stuff like this, we can apply it to ourselves, right? And think about what they said, right? So this is why you need phosphate, because when they break the hydrogen bonds, they need something that can rebond to hydrogen easily. So this is also where nitrogen comes from, and the ability to have it. And this also ties into what they was talking about left-handed. This is why they would want to capture somebody who's left-handed and operate on them and use them, because they have double the bonds. Remember, we saw that left-handed individuals have something called zDNA, which is an isolated DNA structure to them only, and they have double the base pairs we have, and they have less of an intertwined nanometer diameter. So it just makes them easier to do shit like this on. You know, it's just like when you see it together, you can kind of piece together how these fucking evil scientists work. History, let's see. No, I feel like it was something else in here I wanted to show. Yeah, so I wanted to show y'all. I actually wanted to go under. Oh, look at this. See, this is the type of shit I need to look at. Hold on, I haven't even gotten into this. What is eukaryota? I didn't see that they, hold on. Is a phylum of Archaea. It is one of two phyla of Archaea, the other being Crenarchaeota. Euarchaeota are highly diverse and include methanogens, which produce methane and are often found in intestines. Ew. Okay. Anywho. Alright, let's get off this. Off that. It was something else that I wanted to show, and I think it was under function. Hold on for a second. I wanted to show y'all. Somebody mentioned something about nanotechnology, right? Oh, somebody said left-handed people. Let me go back to that real quick, and then I'll jump back into. I want to get into nanotechnology so that you can see how they use. And nanotechnology just refers to, like, it's like it refers to a very small molecular level of genes. So like it's a measurement of atoms in nanometers. That's why you call it nanites and or nanometers or whatever. You know what I'm talking about. Oh, go back real quick. So left-handed individuals, right? Let me just type this in real quick. How did I get to it, y'all? Shit, I forgot how I got to it. Hold on. Hold on. Alright, hold on. I gotta find the breadcrumbs real quick to get to it real quick, because I want to show Goddess that asked this. Well, listen, just know, maybe I can get to it through here. Hold on. Because I know how to get to it. I just don't want to lose where I'm at. Just listen, just know, Goddess, or my Seawig, or my sewing. My bad if I pronounced your name wrong. Just know that basically left-handed individuals, nah, bro, we can't do it like this. Hold on, cause I have to show you. Let's go to double helix, right? Which is gonna show you the helical structures, right? And this is where they should be. No, not there. Okay, my bad. Hold on, y'all. Hold on. Sometimes is it secondary structure, or it might be here. Yeah, no. Secondary structure. No, bro. I just found it earlier. It really irritates me sometimes when is it tertiary structure. There it is. Well, here are the three, right? But I wanted to pull up the chart for you so I could actually show you. Bear with me for a moment. Alright, because I want to there we go. Okay. Okay, so look. There's three different types of, for those who didn't catch this. He said I tried writing with my left hand once it failed. So this is a right-handed person. aDNA. Right? Which looks weird. aDNA looks really weird. That's the structure right there. But this is only when there's some type of adverse effect. So when it's like really, really cold, hypothermia or something like that, right? Then you have bDNA. This is also someone who's right-handed, but this is normal. This is what is normal. These are the three major forms of DNA. Then you have someone who's left-handed, right? Yeah, you say you understand why your dad was forcing you to write with your right hand. Yeah, because they wanted you to conform to society, and also it puts a lot of strain on you when you left-handed because they looking at you. Because you got to understand these things. So when they say helix sense, right, they're talking about helix, which is like it's like your brain senses. I can't really explain it much better than that right now. Just know that has to do with your brain motor senses. But repeating unit, right, this refers to in DNA. Let me try to pull up a hold on, I was looking up tryptophan earlier. I don't want to take that up actually. Hold on. I should have kept one of these up the whole time. This is the one that I used. That's the one I used for this. So like, alright. So that's A. This is aDNA. Right? Where's my link? Where am I at? Okay. So the base pairs refer to this. These, right? Maybe you can see it better when I do this one. Hold on, do this one. Alright. Base pairs are these. That's a base pair. That line, base pair. Right? Those are our base pairs. And base pairs are, they follow a group of rules called base pair rules. You know, fuck their words, alright? But they follow a group of rules. And see how they point to the base pair? So what they're saying here with left-handed folks, individuals, right, is that they have two base pairs. Right? So they have double the bonds. Let's look at a matter of fact, let's do this. We all know what DNA looks like. Let's look at a Z DNA structure. If they will show us. I never even heard of it before I started looking this stuff up. Alright. So this is B and okay. This one, there's a bunch of them, but let's try this one. Oh, they even show it spinning on this one. So look how that look, right? You can kind of see the helical structure, but they have double the base pairs. So that's why it looks all flicked around these parts, because it's double base pairs there, right? And not to mention the reason that it doesn't look like a perfect symmetrical thing like that is because they have a oh, this is cool because they show you bDNA up here. See how that look, and then they show you zDNA. Now see how it's off, but these sections that are closer can send information through faster, right? So I'm just showing you that so it makes sense when I say they have 2 base pairs for every repeating unit. When they say repeating unit, this is a unit, this is a unit, this is a unit, this is a unit. So they're saying repeating unit, right? So they're saying you have 2 base pairs for every repeating unit, right? Then the base pair turn, right? This turns faster. This is how many turns. You remember that one that we just saw circling? It turns faster because of the way. Just wanted to know why you said that they are after left-handed people. Yeah, I mean, it's more so, I'm not trying to say it like they're after left-handed people. I mean, they after all of us. You know what I'm saying? If you got carbon in your skin, they after you. But I'm just saying that they would make a better specimen. You know what I'm saying? Even if I was doing study, they would make a better specimen because the diameter, which is basically, the amount, the line from one, in a circle, the line from one point to another is measured in nanometers. Theirs is only 18 nanometers. So even if you were running tests, so this is 18 nanometers apart versus this one, which is 20. And and the right hand DNA is actually 21 when you are under stress or under some sort of a an issue. And they tell you what that is up here somewhere. They say like aDNA is as a result of extraneous behavior, something like that. But basically, I'm just saying this is the reason why you would want them. Not to mention, look, glycosyl angle, right? Anti-ansin, right? The glycosyl angle just refers to like how well it can bond, and it can do that and this. So it just more is always better. When you're talking about bonding and you're talking about like adding on and DNA replication and all that kind of stuff. Sugar pucker, what is that, y'all? Hold up, y'all. They talking about giving kisses. Let me find out left-handed people give kisses. Pugger up, sugar. A cyclic compound is a term for a compound in a field of chemistry in which one or more series of atoms in the compound is connected to form a ring. Duh, okay. So why'd you tell us that? Okay, so they basically just telling you rings may vary in size from 3 to many atoms. Okay, so they're basically telling you that right-hand aDNA has 3. The normal one has 2, and this one has 2, but for cytosine and guanine, which is the double base pairing, I'm assuming they're referring to. They just talking about the compound structure. Like this is just, one cyclic endo. Endo refers to like, see, a lot of this stuff is kind of it's not hard to explain. Let me not say that. That's casting a spell in itself. But endo means like when you have a circle and it's just in itself. So it's intertwined in itself, right? Like an 8 is an endo, right? In chemistry. Exo is like when it's like circle and then it like branches out to here and then branches out to here. That's exo. So they're saying these are endo, and then this one has one that's exo, guanine. Alright, yeah, if your internet won't stop freezing, I don't know what to tell you about that. Reflection, try to come out and come back in or refresh your connection, you know what I mean? I just wanted to go into detail about that because I want to make sure that I touch base on that. Anytime somebody asks something, I don't want it to be like I'm just like saying shit, like, you know what I'm saying? Alright, so let's go back now. That was me doing my search and shit. So like I went into damage, right? Did I go to damage? Hold on. Bear with me, family. Oh yeah, so here we go, baby. Peep this out, right? listen to this diabolical bullshit. DNA nanotechnology uses the unique molecular recognition properties of DNA and other nucleic acids to create self-assembling branched DNA complexes with useful properties. Self-assembling, bro, that is maniacal. Like, self-assembling, nigga, you want them to make themselves? This is iRobot. This is iRobot. And by the way, for those who like, oh, but that's Wikipedia. Wikipedia makes stuff up. They trying to scare you. Go to this Ruthman PW. I don't know who that guy is, but he made something about DNA folding. DNA folding is just a process that takes place during the transcription process in RNA, which would make sense that he was talking about this because they probably use that to program the nanites to do what they're gonna do. Like, you know what I'm saying? But you can click on this and, you know, and read a little bit further if you like. Alright? Anywho, it's also number 171 in the resources down the bottom, which you can go look at. Now let's continue. DNA is thus used as a structural material rather than as a carrier of biological information. This has led to the creation of two-dimensional periodic lattices, which is just a structure that looks like this. It looks like lace. And three-dimensional structures in the shapes of polyhedra. Nanomechanical devices and algorithmic self-assembly have also been demonstrated, and these DNA structures have been used to template the arrangement of other molecules such as gold nanoparticles and streptavidin. Streptavidin, what the hell is that? A 66 tetramer, which is basically a shape of a molecule in a tetra shape. Protein purified from the bacterium dissociation constant. Okay. Yeah, so that's basically, these are instructions. These are codes that they're giving to the nanoparticles. You understand? Like, all of this is coding. This, anytime you see protein, you know what I mean? And they said they got this from, they took this, they derived this from bacteria. Boy, I tell you. Anywho, so, you know, this basically told us in this paragraph right here, which can be referenced in this citing right here, and any other, oh, this one right here too. This person. Assembling Materials with DNA Guide. This is a science book. Go get that book for all the people who like scared to learn here. Alright? Now, history and anthropology. Because DNA collects mutations over time, which are then inherited, it contains historical information, and by comparing DNA sequences, geneticists can infer the evolutionary history of organisms, their phylogeny of organisms, wait, why does that sentence sound weird? Because DNA collects mutations over time, which are then inherited, it contains historical information, and by comparing DNA sequences, geneticists can infer the evolutionary history of organisms, their phylogeny. Okay, so what they basically saying is they can study this. They can study the DNA that they collected to figure out where people fit in this tree. Phylogeny is a phylogenetic tree that tells you, like, phylogenetic, my apologies. Phylogenetics is the study of evolutionary history and relationships among or within groups of organisms. These relationships are determined by phylogenetic interference methods that focus on observed heritable traits, right? So what's up to you, Reflection Peace family? I appreciate you coming in. Watch this back if you get a chance. Ton of information and things that we went over. We just dissected how they use nanotechnology to have self-assembly, algorithmic, right? Algorithm. This is a set of instructions. That's what an algorithm is. So that means they basically tell it what to do, and then it just does it. Shit is sick. Why would you even want to do that? Anyway, this field of phylogenetics is a powerful tool in evolutionary biology. If DNA sequences within a species are compared, population geneticists, what the fuck, population geneticists? You see how evil that sounds? I'm gonna go back to that because that shit sound crazy. What's up to you, Jessmo85? If you get a chance, watch this back. Sending positive vibes to you. I want everybody to have this information. There's no reason we should know more about the cars we sitting in than the bodies we driving. All right, one love to you. So let's read this back, but I'm gonna go back to this. If DNA sequences within a species are compared, population geneticists can learn the history of particular populations. This can be used in studies ranging from ecological genetics to anthropology. And what's anthropology, y'all? Some evil ass shit. We all know. What is it? What they tell you it is? Let's see what they say it is. Anthropology is the scientific study of humanity concerned with human behavior, human biology, cultures, societies, and linguistics. Let me put that in layman's terms. Anthropology is where we come to where other people are, study how they behave, and study the insides of their bodies and their living organisms, study all of the things that they've been doing throughout their life, how they interact with one another, and how they communicate with each other. That's what they basically saying. We come to your shit and figure shit out. That's like, listen, if they sent anthropologists to your house, I want y'all to see me laugh on this one. Listen, if they send anthropologists to your house, son, that's the hit squad. You hear me? That is the anthropologist, because alright, family, listen. I want to make sure I get this saved and all of that good stuff in my internet look like it's acting a little funny. So look, what I'm just gonna say is that's because I was talking about anthropology. They ain't like that. They was like, he is telling them what we do. Go look at my lecture titled Human Settlements if you want more detail on that. But listen, shout out to everybody who watched this live. If you made it through this live, like, if you made it through the whole live, first of all, one love to you. If you made it through even more than half of the live, sending positive vibes your way. And even those who didn't see it, I just hope that they see it, and I hope that you get any information from this that you can. You know, I feel like I pretty much touched on most of the stuff that I would want to touch on. Oh, let me show y'all something. Hold up. Let me show y'all something real quick. Listen, there's a whole lot of different types of DNA, right? We learned about 3 different types today. Y'all remember? Class? Now I'm messing with y'all, but listen. We got aDNA, which is for what? Right-handed individuals that are experiencing some form of trauma like hypothermia, right? Or physiological ailment. We got bDNA, which is normal, right? And then we got zDNA, which is for lefties, right? Now, damn, I spent so much time trying to tell y'all about that I forgot what I was about to go into. Oh, so listen, there's multiple types of RNA. Right? Like, you know, you have mRNA, right? Which is messenger RNA. You have tRNA, which is transfer RNA, right? But you also have something, right? And this is where I'm about to show y'all what cloning is. That they don't show you about, right? You gotta like dig deep to find this. It's called shRNA. Now, what is shRNA? Let's read up, cause reading is fun and dumb for your mental. One love to everybody. Listen, a short hairpin RNA, aka shRNA, is an artificial RNA molecule. First of all, we don't need to read shit else. Y'all see that? An artificial RNA molecule. But let's keep going. With a tight hairpin turn, right? They just describing what it looks like, right? Because this is shRNA. They just describing it, right? And let's see what it's used for. It's used to silence target gene expression via RNA interference. You see that? Now see, they're not going to show you this because this is not in the realm of like normal DNA. This is in the realm of some diabolical mad scientist type shit that they would know about. I found this because I was just digging, trying to figure out any other kind of DNA and RNA I could find when I found the A, the Z, and the B, or when I found the A and the Z, because I didn't even know that they were multiple, right? Hold on, y'all, my phone about to die. Wait a second. Alright, so look. When you look at this, you see that they tell you expression of shRNA in cells is typically accomplished by delivery of plasmids, right? And we spoke on what plasmids was before, right? Y'all remember? Plasmids was, let's see what they say, right? Plasmids is a small extrachromosomal DNA molecule within a cell that is physically separated from the chromosomal DNA, right? This is viruses. This is viruses, bacteria. And you know that because when you look up virus, it tells you that exact same thing about it. And look, and right after, look, they tell you right after it. Plasmids or through viral or bacterial vectors. You know what I'm saying? Like, and vectors are just coordinates for how they do shit. Look at this. In molecular cloning. And we having debates about if cloning is real. In molecular cloning, a vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell. And see, this is why I was saying all of these scientific things run together. You know what I'm saying? Because vectors are used in sacred geometry. They're actually used in geometry as well, and physics. But vectors, like, determine, like, how much of something you can fit into something else, right? So when they're using vectors, they're using that to determine how much of the plasmids they can send through, you know? So it's like, that's why it's helpful to learn all of it. You know what I'm saying? It's helpful to learn all of it, to just see it. Like, even us doing this today, like, we don't know every single thing, but like, we know a lot. You know what I'm saying? Like, now somebody not gonna just walk up to you and be like, yeah, you know, you don't know nothing about your DNA. You're gonna be like, first of all, DNA stands for deoxyribonucleic acid, where look, shh. Oh, shit. Shh, RNA. Shh, RNA. Oh, that's... Anyway, you're going to be able to break down the phonetic definition of deoxyribonucleic acid, because deoxy simply means the removal of oxygen atoms, right? Ribo refers to ribose, which is what? A type of sugar. And we actually didn't go over ribose. I actually didn't even step into ribose with y'all. But look, if we was to, right, let's go into ribonucleic acid. And then here it should say ribose. Ribosomes, right? And ribose is... Alright, look, see, they irritate me with taking so long. I'm about to just type it in, okay? Because there's breadcrumbs that you follow to get to all of this stuff. Alright, ribose, right? Ribose is a simple sugar and carbohydrate with molecular formula CHO. This is what I was telling you about CHO. This is what Dr. Sebi was teaching me. Yeah, that was deep. That's this real shit. Like, they show you this right in front of your face. Like, I feel like we need to go into RNA further. But I just wanted to show real quick that CHO is what all things see. And they got this, listen, unnatural sugar is L-ribose, right? This is why this is real sugar. Real sugar that naturally occurs is called D-ribose, right? And this is a component of ribonucleotides from a sugar called ribose. Now, what you should be probably looking up is, where do I find ribose? What foods have ribose, right? Now look, I'm gonna take a wild gander and say they gonna tell you to search in some goddamn Dairy products. Oh my goodness. Hold up, y'all. Let me see. Okay, they gave you one. Look, see how they told the truth a little bit. Look at you. They told the truth a little bit. Ribose can be found in both plants and animals. See, they don't usually say that. They normally tell your ass, go get that yogurt, which is cultures of milk. Yogurt is basically bacteria that's been sitting around until it becomes like fungal. Anchovies, herring, sardines, there they go with that bullshit. Caviar, there they go with that bullshit. Eggs, no. But yeah, they definitely got beef and poultry, cheddar cheese and yo, they don't be playing, They be like, you gonna get this cheese. Oh my goodness. They say you gonna get this cheese, boy. Yeah, so eat mushrooms, y'all. Eat mushrooms. Cause on this list of this BS that they talking about, the only thing on here that's natural is mushrooms. And that's the only thing that's plant life on here. That's why they said both plants and animals. See, but they don't tell you more than that though. There's more. There's more than just mushrooms. You gotta look deeper, like, and see, what you really gotta understand is that ribose, D-ribose, right? Make sure it say D-ribose, because L-ribose was made up by them and it's fake. But this is just RNA. So you can actually find this in meat, because meat has proteins, which has RNA, the blood cells. So when you're eating, this is why they say you are what you eat. When you eat meat, people don't understand you are eating RNA. You know what I'm saying? Like, you literally are ingesting molecular instructions that are going to tell your body what to do. You got to understand that. You know what I'm saying? Because look, so I would recommend that people try to look up. You can go to Encyclopedia Britannica too. Britannica's cool, but like I said, Britannica is not going to give you, does ribose raise blood sugar? Bro, how can it raise? Ribose is a naturally occurring sugar, but it doesn't impact. Duh. Why would it impact it? And it's what our DNA made out of. But you know what does impact it? Look. Ribose is naturally occurring. It doesn't impact blood sugar like sucrose, fructose, right? Now, actually, fructose is natural. But fructose is not generated by your body in itself. It comes from your diet. It can be synthesized by your body, but it doesn't like your body doesn't sit and create fructose. You know what I'm saying? That's why it can affect you negatively if you just like overdo it like crazy. And this is why, look, I know I'm going everywhere, y'all, but look, this is why when we say stuff like this, right? When I say fructose chemical structure, right? Look at the fructose chemical formula. 6 carbon atoms, right? Come on, why does my phone keep... Hold up, y'all. I don't know what's up with this junk. 6 carbon, 12 hydrogen, 6 oxygen, right? That's it. That's what you need for fructose. Apple a day keeps the, you know what I mean? Yeah. Right? Now, I just want to see if they're going to pull this up, because I was trying to find this earlier and it was irritating me. There it is. Now, y'all seen me kept trying to find that. That's because they doing the malic acid in it. Okay, that's fine. But it's 465. I don't know, which actually comes back to 6, because that's 15. 4 plus 6 is 10, plus 5 is 15. 1 plus 5 is 6. But, you know, everything come back to the 3, the 6, and the 9. Right? But we saw how low those numbers was, right? Now, let's type in chemical formula of... Let's do What's the one that they always put in stuff? Sucrose. They always put sucrose in stuff. And let's say sucrose. Look at this. Look how much that is. First of all, the apple was 6, so that's double there. That's almost triple there, and that's double again there. That's in sucrose. Sucrose, right? What's the other ones? Right? Listen, we don't gotta do this. I ain't gonna waste y'all time with this. But I just wanna show you, when you look that up, you know what I'm saying? All of the natural sugars have low atomic numbers. But when you look up white sugar, it's damn near 3 times. So that's why you oversaturate in your body. Your body can't do anything with all of that. So this is where it turns into things like fatty acids. You know what I'm saying? This is where certain things, you remember, if y'all remember when we was talking about the non-coding in the RNA, right? Let's go back to it, right? I actually typed this out, so I don't think it's going to come up. But remember how they was talking about how they are non-coding. Look, just keep in mind that there's non-codon instructions that come through, and those are things that can't be coded. It increases those when you got too much sugar, and it prevents you from bonding appropriately. You know what I'm saying? You want to understand something, right? DNA, right? These acids, these nucleic acids, are held together by something called a glycosid. Fuck they words, man. Hold on. Alright, so they're not telling you here, but there is something called a glycinic bond or glycogenic bond. That's what holds them together. I can't find it right this second, but that's what holds them together. Now, in order for that to stay strong, you have to have a certain balance. You know what I mean? Like, you have to have a certain balance of all of these things in your body. You know, the sugars and everything. And this is why when you put other stuff in your body, you throwing this out of whack. You know what I'm saying? You throwing it off. And this is where aDNA comes from, right? We was talking about, right? aDNA. Cause I never explained, like, I didn't go into detail. You know what I'm saying? aDNA is one of the possible double helical. I'm trying to let y'all see too. They messing up my camera. Is one of the possible double helical structures which DNA can adopt, right? So that means DNA changes into this. It's not just that way naturally. It can change into it. aDNA is thought to be one of 3 biologically active double helical structures along with bDNA and zDNA. This is left-handed right here. It is the right-handed double helix, fairly similar to the more common bDNA form, but with a shorter, more compact helical structure whose base pairs are not perpendicular to the helix axis, as in bDNA, right? It was discovered by whatever, who cares about her, who also named the A and B forms. So she named them. Somebody named Rosalind Franklin. Who was she? I'm gonna give y'all a hint. Your ancestors not here because of her. Yeah, right. English chemist, right? An X-ray crystallographer. Blah, blah, blah. So, you know, but there was, what I wanted to say was, oh yeah, she showed that DNA is driven into the A form when under dehydrating conditions, right? Such conditions, come on, bro, fix my camera. Such conditions are commonly used to form crystals, and many DNA crystal structures are in the A form. The same helical conformation occurs in double-stranded RNAs and DNA-RNA hybrid double helixes, right? And they just saying what I was saying right here. aDNA, yo, this is crazy, is fairly similar to bDNA, given that it is a right-handed double helix with major and minor grooves. However, as shown in the comparison table below, there is a slight increase in the number of base pairs per turn, resulting in a smaller twist angle and smaller rise per base pair, making a DNA 20 to 25% shorter than B DNA. The major groove of A DNA is deep and narrow, while the minor groove is wide and shallow. ADNA is broader and apparently more compressed along its axis than BDNA. So that's basically letting you know that the compression, so when things happen like when you don't have enough water, remember what they were saying. This is caused by lack of water. Now, when you drinking a bunch of soda, you know what I'm saying? You see how it all tie in together. Now it makes sense why you should be actually drinking something that's going to hydrate you. Because like she said, and forget her, we don't care about this lady, but, for his story, she showed that DNA is driven into the A form when under dehydrating conditions. So that means a lot of our DNA structure, let's see if they're going to show this. They might show. Look at that. That's what it looks like. See how flicked that is? Oh, twined it up. Now it does have one base pair still there, but the distance is further too. It just makes it so that there's more room for error. That's all. That's really what it's about. There's more room for error. So, and look, this is dated from, this is the title, the author, for those who like, oh, that's Wikipedia, somebody drew that and just uploaded it. You know what I'm saying? You gotta have receipts for Wikipedia for people, man, because they be swearing up and down that everything on Wikipedia a lie. Listen, here's one thing you want to know about Wikipedia. The people who would want to lie on this would not be telling you this. So anytime you looking for this kind of information, chances are it's going to be factual, because why would they get on here and lie and tell you this stuff? Like, if they was lying, they would say stuff like, yeah, there's never been any, like if we was reading and it was like, there's no proof of cloning, I would be like, all right, yeah, somebody jumped on here. You know what I'm saying? But I don't think somebody is going to jump on Wikipedia and make up a lie that's tactful and specific scientifically about how cloning happens. You know what I'm saying? And there's something called subcloning too. Like there's different levels to cloning, y'all. And I wanted to touch on it all, but like I said, I don't want to make it too long. I hope that everybody got something out of this. I would advise you to do your due diligence to research all of this stuff, specifically biochemistry. I'm coming at anatomy next. You know what I'm saying? And there's much more in biochemistry for me to target. Like I can do a whole lecture on specific things like gene expression. I just happened to see the lie was that wiki isn't a good source of it. Yeah. The lie is that you should confine your intellect to one space. You know what I'm saying? Like anytime you're being limited is a lie. Even if it's wrong, so what? Find out. You know what I'm saying? If somebody came up to me and said, yo, it's a bunch of dudes with sticks and knives outside, so they about to break in your house, I'm not going to be like, I don't care because I think you're lying. I'm going to go look out the window. Like, you know what I'm saying? Worst case scenario, he lied. You know what I'm saying? But you said things making sense don't last long with lies. Yeah. Yeah. So, yeah. One love to y'all reflections. Please remember to be positive, optimistic, keep positive people in your circle, and share any of this information with anybody else that you can, who you think might want to know. You can come back to this live, or you know where to look up. After what we just went through, you know where to go to look up information if you got a question about something. If you want to know, like, well, what does this do? You know what the 4 nucleobases are. Which are what? Guanine, cytosine, thymine, adenine, right? And then you got uracil, which is native only to RNA, right? You can look up these things and want to know why. You can also look up splicing. Like, splicing is a real thing too, and I wanted to show y'all that too, because they got a whole section that's dedicated to splicing, where they talk about, you know, how splicing works. You know what I mean? It's called gene splicing, and I just didn't go into it because we pretty much already covered enough. Like, there's nothing else they can say other than, "Hey, y'all, we over here making clones, dog." Like, you know what I'm saying? At this point, that's the only thing they can say. All right. Sending positive vibes to y'all reflections. One love. I get up with y'all later.